Good morning, everyone, and welcome to our virtual public workshop, Statistical Considerations for Digitally Derived Endpoints in Clinical Trials. I'm Marianne Hamilton Lopez, and I am the Senior Research Director of Biomedical Innovation at the Duke-Margolis Institute for Health Policy. I'm excited to welcome you to today's meeting, which is being co-convened by Duke-Margolis and our colleagues at the U.S. Food and Drug Administration in the Office of Biostatistics within the Center for Drug Evaluation and Research. Today, our sessions will focus on the statistical and methodological considerations needed to advance digitally derived endpoints from development and validation through regulatory decision-making. Advances in digital health technologies are transforming the landscape of clinical research. These technologies span innovations such as wearable sensors and mobile apps to ambient home monitors. Digitally derived endpoints stemming from these technologies offer insights into patient health, functional capacity, and treatment response between traditional clinical visits. This is accomplished by capturing continuous data from participants outside of the clinical environment. The FDA has demonstrated a commitment to fostering innovation in this space while ensuring that digital measurement tools meet the highest regulatory and scientific standards. Today's meeting brings together regulators, patient advocates, researchers, technology developers, and clinical trial sponsors to address critical challenges, including algorithm validation, handling missing continuous data, and reducing patient burden. Developing and validating digitally derived endpoints is a collaborative endeavor. It requires bridging the gap across areas like sensor engineering, clinical validation, statistical methodology, and patient usability. We look forward to a robust discussion regarding how to advance patient-centric medical product development and regulatory review through digitally derived endpoints used in clinical trials. I would like to start with a few logistics before we dive into today's content. On the screen is a short note about our commitment to academic independence. You can find more about our policies on this slide. Duke-Margolis is convening this workshop under a cooperative agreement with the U.S. Food and Drug Administration. The conversation today, while supported through a cooperative agreement with FDA, is not a federal advisory committee, meaning we will not be voting, we're not making recommendations, or taking any binding actions. Throughout the meeting, attendees are encouraged to share comments and questions for our moderators, presenters, panelists using the Zoom Q&A function. We do have a large group that have joined us today, so we're going to do our best to incorporate questions from the audience into the panel discussions. If you have any technical issues, please use this email address here for Margolis Events, and our staff will work to address them. And then lastly, because this is our number one question during chats, the meeting materials will be made available on the Duke-Margolis website after the event. Now, I am pleased to present an overview of today's session. We will start the day with opening remarks from Mark Levinson, the Director of the Office of Biostatistics at FDA. Then we will have our first session, Regulatory Efforts to Advance the Use of Digitally Derived Endpoints for Product Development and Review, which will provide an update on FDA's PDUFA VII initiatives and supporting digitally derived endpoints, with discussions of patient perspectives, development trends, and opportunities to improve data standards and submission practices. After a brief break, we'll reconvene for our second session, which is continuous glucose monitoring technical specifications as a model for regulatory submissions, device evaluation, and analytical best practices. Speakers during this session will discuss FDA's CGM technical specifications document and examine how CGM's derived data can be standardized, analyzed, and applied to support regulatory evaluation of digital endpoints with lessons from other digital health technologies. Following session 2, we will break for lunch, then begin the afternoon with our third session, Statistical and Measurement Considerations in Developing Digitally Derived Endpoints for Regulatory Review. This session will examine key statistical measurement and validation considerations for developing digitally derived endpoints, including data provenance, remote monitoring, and analytical and clinical validation. In our fourth session, addressing statistical challenges to analyzing digitally derived endpoints, panelists will explore statistical challenges in analyzing and reviewing digitally derived endpoints, focusing on missing data, regulatory review lessons, and approaches to consistent evaluation. After another brief break, we will begin our last session of the day, focused on future considerations for digitally derived endpoints, where panelists will discuss opportunities, challenges, and emerging innovation that is shaping the development, the evaluation, and regulatory use of digitally derived endpoints. Now, I will turn it over to Mark Levinson from FDA for his opening remarks.
Good morning, everyone. Thank you. It's my pleasure and honor to open this workshop. I also want to thank FDA and Duke-Margolis, the organizers of this workshop, for putting together such a great program. As we heard, we're going to have speakers and panelists from FDA, industry, researchers, and academia, and importantly, patient groups. And they're going to share their knowledge and insights. As we just heard, in this area, collaboration is essential for success here. So I'd like to use my short time here to provide a little motivation and thoughts on digitally derived endpoints, particularly with emphasis on statistical considerations. Next slide, please. So I want to start with a traditional clinical endpoint, the 6-minute walk. This has been used as a measure of functional capacity. We've seen it in studies of disorders of cardiovascular, pulmonary, neurological, and metabolic disorders. It's widely used, and it's basically the distance one could walk in 6 minutes. In a traditional trial, the patient travels to the clinical study site, and the test measures how long they could walk in 6 minutes. I will note it's a snapshot in time. There may be variability in the measure for all sorts of reasons. Perhaps the patient is fatigued from their travel to the site. Perhaps they had a bad night's sleep. Perhaps observers of the test motivate some individuals more than others. Again, it's just a snapshot in time. It's probably not a direct interest to the patients. Patients don't perform this activity routinely in their daily lives. Effectively, this test is a surrogate for functional capacity. Also, we're limiting the available population to those who, and with their caregivers, can travel to a clinical site. This has bearing on the representativeness of the study to clinical practice. We may end up underestimating or overestimating the treatment effect. The question before us is how we can come up with a better endpoint that measures something meaningful to patients that is less variable and less burdensome. Next slide, please. I'm sure everyone in this audience is familiar with accelerometers. They're found in smartwatches, phones, and other devices. These devices record acceleration in the 3 spatial directions. It does this continuously. Using such a device, aspects of the patient's movements can be measured continuously without a patient going to a site. Maybe we could use these data to get more patient-centric endpoints with lower variability and lower patient burden. That's easy to say in practice. In practice, we want to ensure this happens, and that's why we're here today. I will return to some of these challenges in a moment. Next slide, please. There are many examples of digital health technology. Here's a range of examples, and it's not meant to be exhaustive. There may be passive biosensors, which are shown on the left of this slide. Some examples of these biosensors include continuous glucose monitors, and we're going to have a whole session on these as a case example. I've worked in the diabetes world, looking at drugs for the diabetes indication, and one measure that's of interest to patient and clinicians is time in range. And that's traditionally very hard to get from using traditional endpoints, but perhaps the CGM can get that more efficiently and effectively. We have other versions of continuous monitors like ECG monitors and blood pressure monitors. And the last two, we have things that are based on accelerometers, fall detectors, and actigraphy. You can imagine these, the actigraphy could be used to derive an endpoint that might replace a 6-minute walk. On the right-hand side, we have more interactive applications. A patient might use an application, a remote application, to complete a survey to get data on a patient-reported outcome. They may take some key clinical pictures at their home. And they also may even perform tests remotely. All these interactive applications may decrease patient burden and potentially reduce missing data. Next slide, please. Okay, let me return to the accelerometer for a moment. Again, the output is three-dimensional continuous measurements. Let's consider using these data to form an endpoint. I'm going to move from the example to try to replace the concept that's trying to be measured in the 6-minute walk to something a little simpler. I'm going to use as an example here fall detection. This simple example raises a lot of important issues. Next slide, please. Okay, so the question before us is how do we translate continuous three-dimensional data into an endpoint? There's animation here, so could you click one more? We want something meaningful to patients. For falls, which is the example I'm using here, perhaps this is reasonably self-evident. Not falling is good, and patients falling can have serious health consequences. So falls, perhaps this is self-evident, is a meaningful concept. Next click. We also want something that's reliably obtained. Now, there's a lot to unpack here, and we'll hear about this throughout the day. This means the device meets its engineering requirements, and importantly, it measures the clinical event of interest. For example, in the fall example, we want to be able to separate a fall from someone maybe just sitting down. Next, please. We also want acceptable precision. Even if the device is measuring the event of interest in the average, if the data are too noisy, this may create inefficient or trials that are not feasible. Next, please. We also have to understand the missing data and how to address it. Can we tell if a person is not wearing the device or the device is malfunctioning, and how we will address these missing data in the endpoint? Next, please. All this requires statistical thinking and methods and expertise, which is why we're here today. I will say, as I've talked about the 6-minute walk example, this isn't easy for traditional endpoints either. We're not necessarily in a more difficult case. In fact, some of these issues were perhaps never completely thought out or just accepted over time for traditional endpoints. So we're not starting off in a worse situation. Next slide, please. So as you're probably all aware, we have guidance in this area on digital health technologies. It covers at a somewhat high level most of these issues I brought up today. But I also want to point out another series of guidance. FDA has developed a series of guidance on patient-focused drug development. And although these guidances are not directly relevant, designed for this purpose, they are relevant. There's a lot of information here that is relevant, particularly getting information from patients on what's meaningful and creating endpoints. Next slide. So I think we've already heard about the agenda, so I won't go through this again in detail, but we have a great agenda today. Again, we have speakers from a range of backgrounds, FDA, industry people, academia, researcher, patient groups. And as I said, and as in the introduction talk said, this is very much a collaborative effort. And I'm glad Duke-Margolis and FDA has put together this program where we could bring together different people and try to move this area forward. So I think we're in for a productive day. And with that, I want to thank the whole audience for joining us, and let's begin the sessions.
Thank you, Mark. Hello, everyone, and welcome to session one of our workshop. My name is Ryan Robinson, and I'm a physician in the Clinical Methodologies Group in the Office of Medical Policy and CDER at FDA, and I will be the moderator for this session, which is titled Regulatory Efforts to Advance the Use of Digitally Derived Endpoints for Product Development and Review. In this session, amongst other topics, an update on FDA's PDUFA VII commitments to support the development of digitally derived endpoints and regulatory decision-making will be provided. And in addition, a landscape analysis of DHT submissions to the FDA will be presented, and a summary of the docket comments from a recent DHT-related Federal Register notice will be discussed. And with that, I'd like to introduce Dr. Matilda Kam, who's the Division Director of the Division of Analytics and Informatics from the Office of Biostatistics and CDER at FDA, and Beth Kinkuski, who is a Health Science Policy Analyst from the Office of Medical Policy and CDER at FDA. They'll be providing the presentations for the session today. And with that, I will turn it over to Matilda to start us off.
Good morning, good afternoon, everyone. From data to decision, that phrase captures what this workshop is about and exactly what FDA is working to make possible. Digital health technologies are giving us an unprecedented look into patient lives, how they feel, how they function, how they survive, which are captured continuously in the real world outside the walls of a clinic. But data alone isn't enough, and the real work is turning that data into endpoints that are meaningful to patients, reliable, and ready to support regulatory decisions. That is exactly what FDA is focused on. So today, I'll walk you through what we're doing to advance digitally derived endpoints in product development and review. Next slide, please. So first, I'd like to define what is a digital health technology, or DHT. So this definition is from the FDA NIH Best Glossary. So DHT is defined as a system that uses computing platforms, connectivity, software, and sensors for healthcare and related uses. They include technologies that are intended to be used as a medical product, incorporated into a medical product, used to develop a medical product, used to study a medical product, and as well as used as a companion or adjunct to a medical product. Here are some examples of DHT used in clinical trials, and you've also seen some of the examples that Mark mentioned in his opening remarks. So wearables, continuous glucose monitors, mobile apps, which allow researchers to capture real-time data remotely, which reduces the need for frequent clinical visits. We also have, contact sensors that can contactless sensors that could passively monitor patients at home, and while an electronic COA platform replaces paper-based data collection with instant data capture. Next slide, please. So this slide outlines some of the exciting opportunities that DHT brings to clinical trials. One of the biggest benefits is convenience. So patients don't need to visit the clinic as often because data is collected remotely. This is essentially helpful for those who live far away and are having difficulty traveling. It also means that we can include more representative patients in our trials. So people who are typically left out which makes our findings more relevant to the real world. Another is DHT also give us better quality data. So instead of relying on a patient's memory at the clinic visit, we're capturing information continuously in real time, helping us catch changes that we might otherwise miss. They also allow us to measure things we couldn't before, which creates opportunities for entirely new endpoints. And as we are filling gaps in disease areas where validated patient meaningful endpoints have long been absent. Next slide, please. So this slide highlights the 6 key statistical and data quality considerations that are unique to working with DHT data in clinical trials. So unlike traditional clinical data, DHT data tend to be noisier and collected much more frequently, which gives us a richer picture of each patient, but also it creates analytical complexities that we aren't used to dealing with. We are also working with new types of endpoints that don't have established standards yet, which makes validation harder. We'll hear more about analytical and clinical validation in session 3. On the missing data side, the problem is twofold. Sometimes we can't always tell when data are missing, and even when we can, we don't usually know why they're missing. So did the patient forget to charge their device? Were they too ill to wear it? Or we often just don't know. And because the data are rich and complex, the usual approaches for handling missing data don't always apply. So this is still an evolving area. So we will hear more about handling of missing DHT data in session 4. The bottom line here is DHT data opens exciting new possibilities, but it also requires us to think differently about how we design and analyze our studies. Next slide, please. So FDA offers multiple pathways for digitally derived endpoints in drug development. The most direct is through the IND, NDA, and BLA submissions, where sponsors validate endpoints on a per submission basis. For a broader reuse across different programs, the FDA's Drug Development Tools Framework offers 3 qualification pathways: the Biomarker Qualification Program, the Clinical Outcome Assessment Qualification Program, and the Innovative Science and Technology Approaches to New Drugs, or ISTAN. Each of this pathway differs in scope, timeline, and evidentiary threshold, but together they form a growing regulatory infrastructure to support clinically meaningful digitally derived endpoints. Next slide, please. So under the Prescription Drug User Fee Act, or PDUFA VII, FDA made a formal commitment to establish a comprehensive regulatory framework for the use of DHTs in the development and review of drugs and biological products. So, recognizing that DHTs were increasingly being used in clinical investigations without adequate regulatory guidance, FDA structured its commitment along the following. So, we have policy development. FDA published a DHT framework in March 2023 and a finalized guidance that Mark mentioned earlier in December 2023, which deals with the selection, validation, and use of DHTs for remote data acquisition in clinical trials. Next is institutional governance, wherein FDA established a cross-center DHT steering committee comprising of senior leaders from CDER, CBER, and CDRH, including the Digital Health Center for Excellence, to ensure consistent and coordinated review of digitally derived data across the agency. Another is through public engagement. So FDA is committed to convening 5 public workshops to solicit input from patients, biopharm sponsors, DHT developers, and academic stakeholders, ensuring that policy development is informed by diverse perspectives. And lastly, it's also through operational infrastructure. So FDA is invested in upgrading its internal systems and secure cloud technology to support the intake, processing, and review of large volume of DHT generated data submitted as part of regulatory applications. So collectively, this commitment reflects FDA's intent to ensure that digital health evidence meets the same evidentiary standards that are applied to traditional clinical data while providing the regulatory clarity and institutional capacity needed to support the advancement of DHT enabled drug development. Next slide, please. So, in this slide, here are some of the recent FDA guidances and publications related to DHTs. In session 2, we'll hear more about the submitting CGM data and clinical trials technical specification guidance document. I'd also like to highlight the white paper that was issued last week by CDER, CBER, and CDRH. It's the teal one on the lower right-hand corner, which is a key. Can you please go back? Key consideration for the development and use of digitally derived measures for clinical investigation. So this white paper is very relevant to our discussion today. So after the workshop, you might want to look through it and read through it. Next slide, please. So including today's event, there have been 3 FDA public workshops on DHTs. We anticipate the 4th workshop to be held early in 2027, and the 5th workshop would be around August 2027. So it's the same time this next year. Next slide, please. Advancing the use of digitally derived endpoints requires building expertise from within. So FDA is expanding our internal review capacity and developing targeted training materials to ensure that our reviewers possess the necessary regulatory, statistical, and scientific expertise to evaluate DHT submissions. Next slide, please. So currently, FDA is involved in a DHT IT cloud demonstration project. So we are approaching this project in 2 phases. Phase 1 leverages FDA existing Precision FDA flat cloud platform to test the full end-to-end workflow, from sponsor submission of a mock DHT dataset through FDA reviewer access and analysis. Phase 2 then takes it a step further, wherein we expanding to an actual DHT generated data from clinical studies, and will focus on analytical and clinical validation. Both these phases are on track for completion in 2027. So this project is about making sure our infrastructure is ready to receive and review DHT data. Next slide, please. So prior to the PDUFA VII, FDA has funded the following CERSI DHT demonstration projects. So these studies have been completed, and we anticipate publications regarding the studies to be out soon. Next slide, please. So this slide lists some of the ongoing FDA-funded DHT projects. I won't be reading them, but these are the list. So next slide, please. So this is my last slide. So if you would like more information about DHTs for drug development, you could visit our FDA webpage shown on the screen. Thank you.
Thank you. My name is Beth Kinkuski. I am a policy analyst in CDER's Office of Medical Policy, and I will be building on and continuing on Matilda's presentation to talk about some of FDA's efforts in the digital health technology space. Next slide. So I will give an overview of CDER's efforts to track submissions, including digital health technologies. Then I'll talk about our recent FR notice and what our next steps will be. Next slide. One of our PDUFA commitments is to track CDER DHT submissions. So in March of 2023, at the beginning of PDUFA VII, we updated Forms 1571 and 356H to include checkboxes, and we asked that sponsors check the box if their submission includes a digital health technology. And the idea is really the technology and data that both Mark and Matilda highlighted this morning, but it's really relying on industry when they submit their submissions to check the box yes. And when that box is marked yes, it is flagged for FDA to know that it includes DHT-derived data. Next slide. So at DIA earlier this summer, my colleagues Ayub Sullivan and Nora Zhao had a poster where we were able to finally share some of this DHT tracking data. So the poster described the data that we received from March 2023 through December of 2025, and finally provided what this data actually looks like for submissions that include DHTs that have been reviewed by CDER. Next slide. So this is, you are manually checking the box that it includes a DHT. So we are manually reviewing those forms to see if they include DHT. So we have a multidisciplinary FDA review team that is going through those forms and then confirming if a checkbox means that a submission includes DHT-derived data. And our definition of what we are looking for is it could be a patient-reported data, such as an EPRO, and also a little more of what we're focused on today is some of those sensor-based measurements, such as watches, patches, mobile devices, contactless sensors, or continuous glucose monitorings. Many of these submissions are coming in on a recurring basis to continue discussions on a specific IND, NDA, so those are duplicates. But again, many of these did not actually include references to the DHT. They might be administrative changes. They might be unrelated to the clinical trial. So those really aren't within the scope of what we're looking at. Next slide. And surprisingly enough, we've had up till December of 2025, we had 2,318 sponsors check the box yes. And so when our team went through those, they confirmed that 277 of those contained DHT-derived data or a plan to collect DHT-derived data, which is about a 12% rate. Many of those are duplicates, but many of those did not actually include a DHT or were more the administrative submissions that weren't really something that we were looking for. So again, that's about a 12% rate. Next slide. So of the 277 that FDA confirmed included digital health technology, this was the breakdown of INDs, NDAs, BLAs over those 3 years. The data is pretty consistent in terms of total numbers. Next slide. So then when you break it down even further, there were 161, which included an electronic patient-reported outcome. 24 or 9% were the CGMs, which we are here to talk about today. And then 92 were really more the sensor wearables that have a lot of interest. Next slide. So the breakdown of review offices. So a lot of those are in the neuro space, which I think many of us are familiar with. This is the Parkinson's, muscular atrophy. Psychiatry is also included in this group. The next highest group was immunology and inflammation. This includes atopic dermatitis, asthma, chronic cough, sleep apnea. The next highest group was cardiology with pulmonary arterial hypertension, AFib, kidney disease, obesity. Next slide. So this is what FDA's current status is on tracking DHT submissions. We do hope to be able to share this information more widely through other avenues as well, and really to get the message out of where we are seeing these digital health technology submissions, and also to ask you to really think before you check that box so that you are checking it and it is aligned with the types of submissions that FDA is looking to track. If you're not sure, you're always welcome to reach out to us to make sure that we are in alignment because we want to make sure that our data is accurately reflecting what is coming through the door. Next slide. So the other area where FDA has been very active recently is earlier this summer, or spring actually, we put out a docket on advancing the use of DHTs in clinical investigations for drug and biological products. And really, we had heard and knew that there have been some challenges, and we wanted to hear directly from all of you of really where can we take this program and what can we do to support you to increase the adoption of DHTs in clinical trials. In particular, we asked for feedback on guidances and workshops, as those are the PDUFA commitments we have to complete within the next year. That docket closed at the beginning of the summer, and I am pleased to share that I can now give you a highlight of what we saw in the docket. Next slide. So we had a total of 39 commenters provide feedback to the docket from a variety of groups, and I'll just highlight the third row down there, the CROs and technology service providers. A lot of that is the DHT developers that are developing these technologies. We received comments from 12 of those organizations, which is really great to know that they are being very vocal, and we had the opportunity to directly hear from them what we can do to help them. Next slide. So there were a lot of areas for consideration moving forward. We specifically asked about the need for guidance and workshops, and there was a overwhelming response that there is need in both of these spaces. We're here today as one of those workshops on the statistical analysis of digitally derived data. As Matilda mentioned, there's more to come on the guidance front. The docket identified a clear need for regulatory clarity. This may include a need for predictable evidentiary standards regarding validation, need for cross-center consistency, need for clarification on the regulatory environment. There's also a need for DHT use cases. We are working on finding the right balance of transparency and protecting confidentiality of investigational new drug applications. There's a need for improved operationalization and transparency on how DHTs are being reviewed. We also recognize that DHTs can truly make a difference in special patient populations where remote data acquisition allows them to participate in a clinical trial where they might not have been able to otherwise. There were also many more recommendations that we are considering that impact multiple areas of clinical trial innovation, not just DHTers. We hear you, we want to do it all, but we can only take one step at a time. But I am happy to share at a high level what our plans are moving forward. Next slide. So CDER is developing a strategic plan to ensure consistent adoption of DHTs within clinical investigations and to provide clear, consistent communication to external stakeholders. These efforts will reduce uncertainty regarding FDA expectations and support broader adoption of digitally derived endpoints in drug development. We've identified three major themes. The first one is engaging the public and interested parties. This workshop is part of that, and we, as Matilda alluded to, we will hold 2 additional workshops in 2027. We want to encourage FDA listening sessions to discuss general feasibility and other DHTs. And we want to identify opportunities for shared learning through our use cases and then updating our webpage. Next slide. We will expand the operationalization of DHTs in clinical trials. If you have had a chance to look at the 2026 CDER guidance agenda, which came out at the beginning of the year, there is a draft guidance listed there on engaging with the FDA to discuss the use of DHTs in clinical investigations. And this really is also something that we heard in the docket of how can industry and DHT developers engage with FDA. And we really hope that this guidance will lay out what those possible pathways are. We're also looking at improved internal processes and submission tracking. We understand there's a need to be consistent across review divisions and the sharing of experiences. I shared where we are with tracking DHT submissions today. check the box, but we also know that with AI and all of our internal tracking processes, that there has to be a better way to get this data and to ensure consistency. So we're working on that. And then finally, expanding into special populations, we will issue guidance describing best practices and strategies for development of efficacy endpoints, including surrogates and intermediate endpoints for rare diseases. This is a Fedora commitment that is in the works to be completed by September of 2027. And then based on the docket comments, we're also exploring additional guidance considerations in special patient populations. Next slide. DHTs are transforming clinical trials. Remote data collection is reshaping how and where clinical research happens. Patients no longer need to travel to a clinic to provide data for a trial, and it reduces the burden and allows them to continue to participate in clinical trials. Regulatory submissions are evolving. They're increasingly appearing in INDs and NDAs. We've shared in the therapeutic areas where that is most common. But we certainly encourage you to accurately report those when you are completing the forms 1571 and 356H. But FDA is committed to advancing DHTs. We are working through transparency and openly communicating where we are with DHTs. Guidance documents are coming. Workshops and demonstration projects are already underway. And then the DHT steering committee ensures that FDA's approach is coordinated across centers, so sponsors can receive consistent signals regardless of which center is reviewing your submission. Thank you, and I look forward to many great discussions today. And this is just a slide of CDER resources for your reference. Matilda and I both mentioned the DHT steering committee, which is always an available resource, and then the link to our webpage.
So thank you very much, Matilda and Beth, for two excellent presentations. As a reminder, I just want to let the audience know that the video and slides from the workshop will be made available after the meeting. And now I'd like to introduce the remaining panelists that will be joining us for the discussion portion of the session. First, we have Dr. Jeffrey Siegel, who's the Director of Drug Evaluation Sciences in the Office of New Drugs in CDER at FDA. And we have Yuga Xiao, who is a clinical research lead at the Michael J. Fox Foundation for Parkinson's Research. Jeff, I'll turn it over to you first to get us started with any opening remarks.
Thank you very much, Ryan. Really looking forward to this opportunity to engage about this exciting new area. I think one of the key issues that was brought up by the opening speakers that I'll be addressing further in my comments later on has to do with how we determine whether a digitally derived endpoint accurately reflects what really matters to patients, how it measures the way that they feel and function. And the processes that we put in place to determine that are going to be really important going forward and depend very much on what concept is being evaluated. I will be talking about this more later on. The other topic that I'll be addressing that I think is very important to think about is how developers of digitally derived endpoints and pharmaceutical sponsors approach the FDA to find out about acceptance of their tool. And this can come through the IND process where individual pharmaceutical company sponsors approach the clinical review division to get input based on the data that they've determined. In this process, they get feedback, but that process is not a public process. In contrast, the qualification program is another way that developers and pharmaceutical company sponsors can get input on acceptability of their digitally derived endpoints. This is a more public process where the outcome of the submissions are posted on our public website. I'll address that later on in response to specific questions. Thank you.
Thank you, Jeff. Now I'll pass it over to you, Yuga.
Great. Thanks, Jeff. Really excited to be here today representing the Michael J. Fox Foundation, which is the largest nonprofit research funder for Parkinson's research globally. And in my role at the foundation, it's been really, really amazing to hear about all of the patient-centeredness that has been discussed already in the intro session, because at my role at the MJFF, I work really closely with researchers, industry partners, and the Parkinson's community to advance drug development tools that are both more sensitive to change, which is really what we're looking to get to here with digital health measures, as well as meaningful to patients. And I cannot stress this piece enough. And I think this is particularly important in the realm of digital health technology and digitally derived measures, that we consider not only what we can measure with these technologies, but how we really make sure that we are measuring aspects of disease that actually matter to people living with the various different conditions. And it's really imperative and critical to the success of the digital measure development process that the participant input, patient input, should be informing whether we are measuring the right thing in the right population, in the right way, and what really the change that we see in the measure means. And I think we really sometimes focus just on the usability side of getting participant input, but I think we really should be expanding that to collect information and patient input around how frequently the digital device should be used, how the data will be informing the study objectives, and ultimately how getting their input on how to interpret the data results. And I think this is really important because we have seen firsthand in the Parkinson's space that where the patient input or lack of patient input can directly impact the quality and utility of the digital data collected. Matilde, I really like the slide that you showed where there was some unique data considerations. Statistical and data quality considerations are unique to digital data, and the bottom three are all related to missing data. And that is really a key piece where we have seen in studies that have been conducted in Parkinson's where lack of clarity around when participants should be wearing the DHTs, especially as it relates to medication, can lead to data that is ultimately very challenging to interpret and have to get tossed out in the analysis. So really getting clarity in patient input is really key. And another example that we often see is digital vendors come to us and they want to be using their device for as long as possible in participants. And so being really clear both to as part of study design and in communication to participants around, okay, what is realistically, how much data can we realistically expect to collect, and conveying the importance of that for statistical power, for quality of data directly to the participants can increase compliance and their buy-in into the study as well. So maybe as we go along in the workshop today, I really urge all of you as participants to think proactively about how participant input and feedback can be solicited across the data collection and analysis process, and not just as it relates to just the participant usability side, which I think sometimes we over anchor to. So I'll just pause there and excited for the rest of the discussion as part of this panel.
Thank you very much, Yuga. So we'll use the rest of the time for open discussion, and just as a reminder to the audience, you can submit questions through the Q&A function. So the first question I have for the panel is, how are patients currently engaged in the development and validation of digitally derived endpoints, and how can they continue to engage in the process?
So maybe I can jump in here, Ryan. I really want to highlight a study in the Parkinson's space that I think has done this particularly well. This is a longitudinal digital study called Watch PD that was industry sponsored but has collaborated broadly with the Critical Path Institute to solicit feedback from FDA and other stakeholders across the development process, which included feedback from FDA to include a qualitative study as part of the overall digital work. And I highlight this example in particular because it not only goes and tries to collect what sort of digital tasks that are being collected and map them directly to how is meaningful. There's this really exciting, interesting methodology, symptom mapping methodology that is used to connect the tasks that are being participants are asked to do as part of the study to directly how patients interpret the meaningful and relevance and importance of those tasks. So I think that's a really exciting study in that sense to be able to directly connect and map what is meaningful to participants directly to what is measured in the study. It's also really exciting because the study group has also included focus groups throughout the study design to provide participant input on how the data that is generated from the qualitative study, from the digital analysis, in a way to help ensure that the results are interpretable to the participants and that they have a first row seat in terms of being able to help interpret that data. And so I think this is a really exciting study for folks to check out if you're not familiar. The Watch PD study in Parkinson's is a really great example for how participants can be brought in across the development process.
Thank you very much, Yuga. We have another question here. What have been the most significant regulatory milestones or policy developments under PDUFA VII that have advanced the use of DHTs in product development and review?
Maybe I can start off our discussion of this one. As I think everyone knows, FDA approves drugs based on the way that they impact the way patients function, feel, or survive. So in developing a digitally derived endpoint, it's important to understand how output from the digitally derived endpoint reflects the way a patient functions, feels, or survives. I think a key milestone in addressing the issues of PDUFA VII was developing a framework for how we map outputs from digitally derived endpoints onto the way patients function, feel, and survive. In some situations, it may seem obvious how the outputs would reflect the way patients function and feel and survive. For instance, a DHT that measures falls, clearly falls are clinically relevant to patients. But if you think about step counts, for example, you would think that in a debilitating disease that effective treatment would increase step counts, but it's not always true that that happens. For instance, in an arthritic condition where patients can't sit still for long periods of time because they get stiff and ache, an effective treatment may relieve their pain and they may actually decrease their step counts. So the improvements in function may be balanced by a better ease at sitting and doing stationary activities. So it's always important to directly measure how digital health technologies reflect the way patients feel and function. The framework that we have adopted for assessing this is based on the patient the PFDD guidance series, patient-focused drug development series, guidance documents 1 through 4, which specify how to assess the way a condition or disease impacts patients and how to determine whether an instrument reflects the way patients function and feel. So in assessing digitally derived endpoint, it's important for developers and pharmaceutical companies using that instrument to consult the patient-focused drug development guidances to determine how outputs reflect the way patients function and feel. And this framework has allowed us to develop a common framework between patient-reported outcomes and clinician-reported outcomes, and now the new area of digital health technology-derived endpoints.
Thank you, Jeff, for that important framing around patient-focused endpoints. As Matilda alluded to, there's the PDUFA VII DHT framework that has outlined what our commitment and plans are throughout 2027. But I can promise that just because PDUFA VII ends, we're not stopping. We're going to continue to push forward and continue this work through PDUFA VIII. The 2023 Guidance Digital Health Technologies for Remote Data Acquisition was a real milestone that really set the stage of how to include your DHT in clinical trials. But I want to just share that our work is not done there. As Matilda mentioned last week, we had the publication of key considerations for the development and use of digitally derived measures. This just reflects our ongoing efforts. We've made some progress, but there's more work to do in this space.
Thanks, Beth. We have a couple more questions. I think we jump ahead to one of them. I think this one was maybe more for you, Matilda. How has the volume and complexity of DHT submissions changed over time, and is FDA's review infrastructure keeping pace with that growth?
So Ryan, like from the presentation of Beth, we've seen that there has been a steady increase in DHT related submissions received by the FDA. Now, this is true not just for the IND, NDA, BLA space, but as well as our various drug development tool or DDT qualification programs. So if you are interested to learn more about the DD submissions we've received, you can go to the CBER Drug Development Tool Qualification Project Search website, because based on the 21st Century Cures Act, FDA is required to publicly make available any information with respect to any DDT qualification submissions. So to prepare for all this increase in and also anticipated increase in DHT related submissions, FDA is scaling up our reviewer capacity. We're developing best practices documents, as well as building specialized training to equip our review staff, with regulatory, statistical, and scientific competencies required to assess DHT-based submissions.
Thank you, Matilda. Looks like we're nearly at the end of our time here. We do have quite a few more questions, and I'd let the audience know that staff and other subject matter experts are going to try to address those questions as best as possible offline. So I just want to thank the presenters and the session panelists for an excellent and very informative session. And now we'll have a short break before reconvening for session 2.
Hi, everyone, and welcome back. I'm Jessie Lynn Dunn, Associate Professor of Biomedical Engineering and Biostatistics and Bioinformatics at Duke University. I'm excited to moderate our second session. In this hour, we'll explore FDA's technical specifications for submitting CGM, or continuous glucose monitoring data, in clinical trials, including application to regulatory submissions and best practices for CGM-derived endpoints. I'll first introduce our presenters. First off, we will have a presentation by Yoon-Hee Kim, lead mathematical statistician in the Office of Biostatistics within the Center for Drug Evaluation and Research at the FDA. Next, we have Hye-Soo Cho, a statistician in the Office of Biostatistics within CDER at FDA. Then we will hear from Yi-Duo Wu, technical expert in the Office of Health Technology within the Center for Devices and Radiological Health at FDA. And finally, we will wrap up with a presentation from Irina Gainanova, Associate Professor of Biostatistics at the University of Michigan. Yoon-Hee, I will pass it over to you to get us started.
Thank you, Jesselyn. Good morning, everyone. I'm Yoon-Hee Kim, a statistical team lead for anti-diabetic and obesity products in the Office of Biostatistics at CDER. Today, I'm happy to present FDA's technical specification guidance on submitting continuous glucose monitoring, or CGM, data in clinical trials. Next. Here's what I cover today. Brief background on diabetes and CGM technology, followed by the core of the guidance, SDTM and ADAM specifications for CGM data, and concluding with a few key takeaways. Next, please. Diabetes is characterized by high blood glucose due to insufficient or ineffective insulin. Type 1 diabetes, which is about 5 to 10% of cases, involves inadequate insulin production. Type 2 diabetes, which is 90 to 95% of cases, involves insulin resistance. The public health burden is substantial, and evaluating therapies for this population rigorously is essential. Next, please. CGM has become central to both clinical care and clinical research. CGM is a wearable device that measures real-time blood glucose continuously. Approximately 40 to 50% of type 1 patients currently use CGM, and uptake in type 2 is growing rapidly. The ADA recommends CGM as part of standard diabetes management. From a regulatory standpoint, CGM-derived endpoints are now commonly used to evaluate both efficacy and safety in drug submissions. Next. Most CGM systems have three components: a sensor inserted under the skin, a transmitter that relays data wirelessly, and a display device or smartphone application. All currently approved and cleared CGM devices are tracked in Devices at FDA database. Next. Now turning to the guidance itself. As CGM-derived endpoints have become increasingly common in regulatory submissions, FDA recognized the need for a clear, standardized framework for how this data should be submitted. The guidance provides specifications for CDISC-compliant SDTM and ADAM datasets, giving a standardized pathway to submit CGM data in support of drug or biologic marketing applications. Next. I want to take a moment to explain why this guidance matters beyond just formatting requirements. CGM datasets are inherently high volume. A single device generates readings every 1 or 5 minutes across weeks or months. Without consistent structure, rigorous data management and traceability simply aren't feasible. Every aggregation step, from raw epoch level readings to final endpoint summaries, must be auditable. Inconsistent submissions can obscure data quality issues and undermine the scientific defensibility of trial results. That's what this guidance is designed to prevent. Next. CGM data has a multi-layer hierarchy as shown in this pyramid illustration. At the base, there are epoch-level readings, which are individual glucose values measured every few minutes. These aggregate into daily summaries, then into weekly or monitoring period summaries, and finally into endpoint level statistics at the top, like a timing range or coefficient of variation. Each layer builds directly on the one below it, and this hierarchy must be preserved and traceable throughout the submission. Next. So let's dive into the guidance. The guidance is organized around two complementary CDISC standards. SDTM, the study data tabulation model, captures raw tabulated data as collected. ADAM, the analysis data model, builds on SDTM by adding derived variables and analysis flags, making the data directly usable for endpoint evaluation. Together, they allow analysts to trace any summary statistic back to its source data. Next. For the next few slides, let's look at each part in detail. Epoch-level CGM data should be submitted in the SDTM laboratory test results, or LV, domain. A key requirement is documentation of missing data. Missing epochs must be categorized and explained not simply absent. Common reasons include device warm-up, device errors, and sensor removal or non-wear periods. Notably, non-wear periods generate no rows in SDTM, but must still be accounted for in ADAM dataset. Next. Beyond epoch-level readings, submissions must include the FDA-required unique device identifier linking each reading to a specific device plus system and sensor-level metadata. We strongly encourage using the same CGM model and version throughout the trial. Switching devices mid-study introduces variability that complicates interpretation. The disposition dataset must also capture any CGM discontinuation events. Next. At the ADAM epoch level, the dataset expands on SDTM by adding analysis-ready variables, including visit assignments using full timestamps and indicator variables for pre-specified criteria such as flagging hyperglycemia above 180 mg per deciliter. Critically, phantom records are added for missing epoch level values, using empty values to provide a complete picture of the planned wear period and enable systematic assessment of missingness. Next. Above the epoch level, intermediate aggregation datasets, which are by day or week, may be submitted. The final analysis dataset aggregates these into endpoint-level summaries by analysis visit, and these are the primary dataset for evaluating trial outcomes. Study metadata and the source code implementing the pre-specified aggregation algorithm from the Statistical Analysis Plan, or SAP, should be provided to support traceability. Next. Missing data handling is one of the most consequential analytical decisions, and the guidance addresses it directly. Completeness thresholds should be pre-specified in the SAP. For example, at least 70% or 90% valid data per day or week, depending on the context. Criterion flags such as CRIT1 indicate when a participant's data falls below that threshold. A sequential imputation framework is recommended from epoch level through to final endpoint level. And sensitivity analysis under alternative missing data assumptions should be pre-specified and submitted. The table on this slide illustrates an example final analysis dataset showing time in range by visit with a flag at week 2 indicating that the data did not meet the 70% threshold. Next. Last but not least, the paired glucose analysis dataset links CGM readings with non-CGM measurements within a defined time window, typically 5 to 30 minutes. This pairing enables assessment of CGM accuracy and confirms whether glycemic events captured by CGM are consistent with non-CGM data such as fingerstick, providing a critical check on data reliability. Next. To close my talk, here are key takeaways. CGM datasets are enormous and granular. Consistent standardized submission structure is required for efficient analysis. Standardization is not a bureaucratic formality. It is a prerequisite for sound regulatory science. CGM data has a multi-layer structure that must be preserved and traceable from epoch-level readings to final endpoint summaries. SDTM and ADAM specification provide a standardized framework for this. Missing data at every layer must be documented, categorized, and handled through a pre-specified framework. Documentation for traceability, including source code and study metadata, is strongly recommended. and engage FDA early. Discussion for device selection and analysis plans, including aggregation algorithms and missing data handling methods with FDA as early as possible, is essential to avoid issues later in development. Thank you very much for your attention. Now I'll pass it to Hasu Cho for presenting lessons from review experience.
Thank you, Uni. Hello, everyone. My name is Heisu Cho, a statistician at the FDA. Today, I'd like to share a few insights and recommendations that you may find useful when preparing a CGM application. Next slide, please. Here is the brief outline of my talk. I will start by discussing the CGM data structure, then I will walk through the data integrity check process and highlight a few common issues using examples. After that, I will share an example of how to handle missing data. And finally, I will wrap up with a brief summary. Next slide, please. As Uni mentioned earlier, CGM data structure is a hierarchical. First, the SDTM-LB dataset contains the granular epic level data of raw CGM data. Next is the ADCGM dataset, which is the analysis version of epic level data. It mirrors the LB datasets but adds demographic characteristics, flag variables, and analysis visit, a visit. Finally, the ADCGM dataset is the summary level dataset, which contains the CGM-derived endpoints. Next slide, please. To illustrate this, I've created a typical and hypothetical CGM data example here. In this trial, a CGM device measures glucose value every 5 minutes for all subjects. Data was collected across multiple periods, including baseline, intermediate time points, and a primary assessment time point, for example, the last 4 weeks of the trial. The drive efficacy endpoints here is time in range, TIR, during those last 4 weeks. This is calculated as the number of CGM readings within the 70 to 180 milligram per deciliter range, divided by the total number of CGM readings in that period, multiply by 100 to get a percentage. Next slide, please. Here is what the data might look like in our actual submissions and how we check traceability across data sets. In the LB dataset, you can see the epic level data measures every 5 minutes. If the value is missing, the reason for the missing data is recorded here. Moving to the ADCGM dataset, we have the same information, but now the A visit information has been populated. These visits are assigned based on the visit window that should be strictly pre-specified in the SAP. Finally, based on the ADCGM EN dataset is derived. This aggregates the information to show the total number of readings, the number of reading in range, and the time TIR percentage. For example, subject 1 has enough measurements to meet the missing data criteria, so their analysis eligibility flag is yes. Subject 2, however, fell below the eligibility criteria, so their flag is no. Next slide, please. Based on my experience reviewing the CGM applications, I want to highlight 3 common data integrity issues. First is A visit mismatch. CGM measurement times are saved in hours, minutes, and seconds. When assigning an analysis visit, the full timestamps must be considered. In the table below, you can see an example where the last minutes before the midnight was ignored, resulting in a missing A visit. This unintentionally caused the records to be excluded from the final analysis dataset. So our advice is a visit should be assigned to the old record based on the exact time the reading was taken, relative to each subject's visit start and end time and date as defined in the SAP. Null A visit values will lead to unintended data exclusions. Next slide, please. Second issue is the inconsistent time intervals between records. CGM devices are supposed to record at exact defined intervals, usually every 5 minutes. However, across several applications, we have noticed intervals of less than 5 minutes between readings. This might be due to the device system errors during time shift, but the exact cause is often unclear. So please plan early on for how to handle time changes or prevent those cases. If inconsistent readings do occur, the reason for this error should be clearly documented and included in the submission. Next slide, please. The third issue, unclear number of days per period. Suppose the 70% completeness cutoff is applied for the last 4 weeks, which is 28 days period. This requires the number of reading is typically, the required number of reading is typically calculated as 70% times 288 daily measurements times 28 days. However, as you can see this example below, some subjects have more than 28 days of measurements in that period. And if they were included in the analysis, it becomes unclear whether they are actually met the 70% thresholds for the intended 28 days, or if they only met it because those extra days of readings. So our advice is the acceptable visit window size, specifically the number of CGM measurement days per period, must be clearly pre-specified in the SAP. Next slide, please. Next, I'd like to share a brief example of handling missing data, drawing from the FDA CGM guidance. Here is the SDTM-LB dataset, where the CGM data was measured at 8:37 a.m., but then went missing until 4:35 p.m. In the AD-CGM dataset, we populated the Phantom record to represent the row for the missing epic level data that aren't present in the LB dataset. Next slide, please. Based on this dataset, we generated heat map indicating how many CGM readings were present per subjects per day. In this hypothetical example, the top heat map for treatments A and B are based on the original dataset, and we then explore epic level data interpolation using the CGM Analyzer R package. I'm not going to discuss what data interpolation method I use because FDA does not endorse any particular software or method, and many valid methods exist. And as a sensitivity analysis, we summarize the timing range before and after data interpolation, epic level data interpolations. And as you can see, the results are highly comparable, which demonstrate the robustness of the analysis. Next slide, please. So to summarize my talk today, data integrity and pre-specification are key. Visit window, visit assignment rules, and missing data criteria must be detailed clearly in your SAP. This prevents arbitrary data exclusions and ensure your data sets are reproducible. Second, documents missing data. Always record the reason for missing data at epic level so that we can accurately assess its impact. Use robust imputation method and conduct sensitivity analysis to ensure your results are reliable. Finally, FDA recently released our official CGM guidance. I highly encourage you to refer to the documents to ensure standardization, maintain full traceability, and minimize data integrity issue in your submissions. This concludes my presentations. Thank you so much.
Good morning, everyone. Can you all hear me?
Yes.
Okay, thank you. I'm Ido Wu from the Diabetes Branch at FDA Center for Devices and Radiological Health. Today, I'll walk you through how CGMs are reviewed as medical devices. Next slide, please. I have no conflict and some quick disclaimers. Next, please. He has already shared what a CGM is. Basically, it is an implantable sensor that can measure continuously at every 5-minute interval, usually. Here's an example of CGM trace. Unlike a finger-stick blood glucose meter, which gives you a single snapshot, a CGM gives you a lot more information, frequent measurements, trend information showing how fast glucose is rising or falling, and alerts that warn users before glucose reaches a dangerous high or low threshold. Next slide, please. CGM technology has come a long way since the first 3-day sensor for retrospective data review was approved in 1999 as a Class 3 medical device. In 2016, FDA approved the first CGM for non-adjunctive use, which means it can be used to make insulin dosing decisions as a replacement of finger-stick blood glucose meter. We'll talk more about what that means later. Then in 2017, FDA authorized the first factory-calibrated CGM that does not require finger-stick calibration. In 2018, we accomplished a key regulatory milestone by creating the integrated CGM or iCGM pathway as a Class 2 medical device. iCGM authorization also establishes specific performance requirements for CGM accuracy known as special controls. Most CGMs currently on the market are cleared as iCGMs. Next slide, please. As a tissue contacting medical device, CGM has to undergo a series of bench testing to ensure safety and functionality. Human factors is also important as CGM users have to interact frequently with their user interface, understand glucose information, and take proper actions. But for this audience, I like to focus on the design and review of clinical studies for the evaluation of CGM accuracy, alert performance, sensor life, and data transmission reliability. Next, please. For the pivotal study, which is equivalent to a phase 3 drug trial, the study population should be representative of intended users, balanced for age, sex, and BMI, with sample sizes typically between 150 to 400 adults. Each sensor insertion site, being arm, abdomen, or buttock, should be evaluated separately. There are special pediatric protections. No venous sampling should be done under the age 6, and no glucose manipulation should be done under age 12. Participants attend clinic sessions at the beginning, middle, and end of the sensor wear period. Day 1 is especially important because of acute foreign body response, which can temporarily affect accuracy. During sessions, venous blood is drawn and measured on a lab analyzer to compare, usually every 15 minutes. And more frequent blood draws can be done when glucose is below 70 milligrams per deciliter or above 250 milligrams per deciliter. To cover the full measuring range of 40 to 400 milligrams per deciliter for typical CGMs, glucose excursions are deliberately introduced in using insulin and carbohydrate under strict safety protocols in the in-clinic session. Next slide, please. For point accuracy, the special controls specify the minimum thresholds of the percentage of iCGM readings within a certain range of the comparator. All thresholds apply to the lower bound of the one-sided 95% confidence interval. The thresholds are different for different glucose ranges, as the risks of CGM inaccuracy vary depending on glucose levels. Here, I also want to highlight the key difference in the threshold requirement for less than 70 milligrams per deciliter. Although it appears that the bars is numerically higher for less than 70, with at least 85% agreement within 50 plus minus 15 milligrams per deciliter, it is important to point out that this plus minus is 15 milligram per deciliter, not 15%, which means that it allows for a larger relative difference between the ICGM and the comparator in the less than 70 range. With this is because, for one, CGM accuracy is most challenging in the low glucose range, and two, it is more straightforward to communicate absolute bias at those low glucose numbers. Next slide, please. One important distinction I want to mention is that non-adjunctive use CGM does not mean that the point value of an ICGM is as accurate as finger sticks. If you remember the ICGM special controls from the previous slide, and compare to the requirements for over-the-counter blood glucose meters in our guidance, as shown on the left here, you can see that meters have a much higher bar for point accuracy. The gap in point accuracy is also illustrated in the sample ICGM and meter performance tables on the right. Next slide, please. As mentioned in the special control slide, CGMs are generally less accurate in low glucose ranges. One possible reason is greater susceptibility to noise with low glucose signal. Here we look at two recently cleared ICGMs from different manufacturers. Both exhibited noticeable negative bias in the less than 54 milligram per deciliter range. The mean absolute relative difference, or MARD, is also the greatest in this range. We should keep this in mind when using CGM data to assess outcomes related to hypoglycemia. Another point to note is that different devices may have different performance in the low range. The observed differences in the table could be due to random study effect, but there could also be design differences as manufacturers may decide to tune their algorithms differently, making them more prone to show bias in a certain direction. These differences are an important consideration when using CGM to collect glucose data for drug trials. As Yunhee has mentioned, using a single CGM model throughout the trial may be helpful to avoid issues caused by these differences in devices. Next slide, please. For trend accuracy, the special controls are intended to prevent the CGM from displaying extremely discordant trend information against the true rate of change. The rationale is that if true blood glucose is rising or falling very fast at a rate of greater than 2 milligrams per deciliter per minute, the ICGM should not display a notable change in the opposite direction of more than 1 milligram per deciliter per minute. In this example, the numbers highlighted in the top right and bottom left corners confirm the device met the 1% discordance threshold for directionally opposite rate of change errors. Next slide, please. Alarm performance is evaluated using a true alarm rate, false alarm rate, correct detection rate, and misdetection rate terminology. True alarm rate is the percentage of glucose alarms that are confirmed to be true by the comparator blood glucose, usually within a plus minus 15-minute window of the alarm. In contrast, false alarm rate is the percentage of alarms not confirmed by the comparator. Correct detection rate looks at the percentage of confirmed low or high blood glucose events that are correctly detected by the CGM at each threshold. Conversely, missed detection rate is a percentage of confirmed low or high glucose events that are not detected by the CGM. There is an inherent trade-off here. Take the low glucose alarm as an example. Lower threshold reduces alarm fatigue but may miss more events. Higher threshold catch more events but increase alarm fatigue. FDA reviews this trade-off very carefully in device clearances as it is important for real-time CGM. Next slide, please. As a digital health tool, it is important that CGMs provide data with the expected cadence, usually every 5 minutes. However, there are various reasons when CGM values are not available, such as during the initial sensor warm-up, when wireless data transmission is interrupted, when the algorithm decides to blind sensor readings due to noise, or even to terminate the sensor session early if it cannot recover from noise. Sometimes sensor may fall off the skin due to adhesive failure. One of the special controls is that an ICGM does not allow clinically significant data gaps. Here is a table showing the summary of data availability rate for all sensors used in a clinical study throughout the 15-day wear period. Next slide, please. Related to data availability, we also review data to support how long a sensor can last by looking at the survival rate. As discussed, factors affecting survival could be algorithmic or physical. On the right is a summary of data sensor survival rate data from the same pivotal study as the previous one. Next slide, please. In summary, we created that CGM regulation for interoperable CGMs, establishing performance requirements via special controls and class 2 devices. iCGM clinical studies are designed with sample sizes intended to meet the confidence interval requirements and to be representative of the entire intended use population. CGMs are authorized as a replacement for finger-stick glucose based on the benefit-risk considerations. However, the expectations for point accuracy of CGMs are generally lower than the performance expectations for SMBG, especially in the low glucose ranges. Thank you very much.
Hi, everyone. My name is Irina Gainanova. I'm Associate Professor at the Department of Biostatistics at the University of Michigan. I'm very excited to participate in this session as academic statistician who has been focusing on best practices and statistical methods for CGM data. I have no conflicts of interest. All opinions presented are my own, and they do not represent the opinion of University of Michigan or the National Institutes of Health that funded some of this work. Next slide, please. Here, I provide a typical workflow on what happens when we collect CGM data. We have specified window, then we do some data processing and compute CGM-derived endpoints. So common ones displayed here are time in range, or TIR, mean glucose, glucose variability represented by coefficient of variation, and glycemic episodes. In my talk, I will cover 3 parts of this pipeline. The first one is the window length over which we compute these endpoints. The second is missing data consideration. And third is the software that is used for endpoint computations. Next slide, please. So here are the 3 points that I will cover, and I'm connecting it to the guidelines provided by the FDA. The guidance is clear in that the trial duration of the window should be specified in advance, that the missing data should be reported, and the reasons for missingness should be provided. And third, that the source code for the software should be provided as well. I will elaborate on my views on what is the rationale for some of these considerations, and I would also make some recommendations that the FDA guidelines do not cover. Next slide, please. So starting with the trial duration, the most cited and common guideline comes from the International Consensus Statement of 2023 by Badalino and co-authors. And the guidelines stay with a quote that the confidence interpretation of CGM metrics requires 14 consecutive days of CGM data with at least 70% of data collected, which is predictive of glycemia for 3 months. So the first point to highlight is that the target is to represent a period of 3 months. Why is this a target? There is a large body of evidence for hemoglobin A1C, the standard blood biomarker of glycemic control, and connecting that biomarker over long-term diabetes outcomes. Thus, we are trying to represent the same period of time that A1C covers because it covers roughly 2 to 3 months of average glucose levels. Now, second, why does the recommendation says that 14 consecutive days of CGM data with at least 70% of data collected? This has 2 reasons. One is logistical. For most common CGM meters, 14 consecutive days with at least 70% completeness can be covered by just 1 meter application, so that makes it logistically much easier for the trial. 2, several studies have shown that there is a pretty high level of agreement, as measured by R-square, between 14 days and 3 months of metrics. However, there has been no studies that looked what is the effect of the substitution on the downstream analysis, in particular in the power of clinical trials, if only 14 days are used instead of 90 days. Next slide, please. Here, I would like to highlight our recent work that has been published in Diabetes Care, and the main conclusion that I want you to take away today is that if you're only using 14 days, you're risking to underpower your studies. Specifically, we found that if you look at the 14 days with 70% completeness for the most common metrics, such as time in range, you may need 20% sample size increase compared to the 90-day window. For some other metrics, such as time below range or time hypoglycemic, as well as glycemic variability, the effects were significantly more pronounced. We provide specific adjustments in this paper for the sample size considerations if you have logistical reasons for why the trial should be shorter to make sure that the loss of power is minimized. We also found that the gaps of missiness have an effect under the same percent of data completeness. If you have large missing data gaps, the effect will be more pronounced than if you have epoch level missingness. Finally, our recommendation here is to plan for at least 30 days of CGM data connection to minimize the loss of power. Next slide, please. Coming to the second point of missing data, here I summarize some common patterns of missingness that can be observed, and I also show a visual representation of one subject CGM data via the lasagna plot. Here, each row corresponds to 1 day of CGM data. The color represents the glucose levels, and the gray represents periods of missingness. Short periods are epoch-level missingness. Those are typically safe to assume are missing at random. Large periods are typically sensor replacement gaps, and those could be informative. If it is an unblinded CGM study and a subject is not wearing CGM for a prolonged period of time, it is quite likely that the glycemic management during that period is not so good. Similarly, early discontinuation in certain cases could also be informative, such as in inpatient settings. Because the type and reason for missingness affect what may be the best approach to handle it, I believe this is the rationale for why FDA guidelines specifies that the type of missingness should be reported and the thresholds of missingness should be specified in advance because it may affect their analysis and conclusions. Next slide. To give you one illustration, here I'm looking at the censoring as one particular example of missingness. So most CGM have a measurement range of 40 to 400 milligrams per deciliter. Some over-the-counter versions like Stellar are even more restrictive. Here I give you a real data from an example subject who has type 2 diabetes, and you can see a lot of missing data gaps, but you can also see a lot of breaks at 400 milligrams per deciliter because this particular subject had very high glucose values that were censored. This is not very common, but it's not uncommon in my experience. And I have seen such data for subjects with type 2 diabetes in particular. And the choices that you have may depend on what is the endpoint. For instance, if the endpoint is time and range, then replacing those values with just the threshold, 400 milligrams per deciliter, may still give you the accurate time and range. However, if you do replace all those values with that threshold, your variability endpoint will be attenuated or biased downward. On the other hand, if you do a complete case analysis and you ignore this censoring, then your time and range will be biased upward, but your variability will probably be better than the previous approach. So the most appropriate approach of how to handle missing data in this case may actually depend on the endpoint that you would like to consider. Next slide. Of course, imputation can be done for the missing data, and one of the most common approaches is linear interpolation. The two figures left and right show the original data and linear interpolated data within 45 minutes. Most software by default will compute most metrics on the data that is not imputed, so that's where this step will have to be done in advance. If there is an imputation, the most common approach, again, is to do some kind of linear interpolation over short periods, for instance, 45 minutes or 1 hour. And there has been actually very little evidence that more advanced imputation approaches over short window will do a large difference to most of the CGM-based endpoints. However, I would caution over interpolating over a large window. Glucose is affected by many different factors. It's notoriously very hard to predict over long-term periods. And it's important to note whether the software that you are using or the processing has a limit on the gaps over which it will do imputation. to avoid biased conclusions. And then it's also important to note that for certain metrics, such as episodes that I'll come to next, the interpolation or some kind of imputation has to be done for any kind of viable computation of that metric. And different software, again, may do it differently under the hood. Next slide. So coming to my third point, which is the software, I already have alluded to the fact that software may differ in how they handle missing data and how they handle some metrics. And I want to do even a stronger caution that software choice matters a lot. As a primary developer and maintainer of our package iGlue for CGM data, I know firsthand which steps go into computation of each of the CGM-based endpoints. And I've also worked with other CGM software developers to understand where exactly we disagree and why we disagree. I found that sometimes we disagree even on the simple metrics, like even times below range. There's sometimes minor disagreements. And sometimes we disagree a lot, and that's coming back to the episodes because the clinical definition itself is ambiguous. Main reasons are measurement error differences. So as has been alluded, if measurement frequency is unexpected, some software may not handle this properly. Missing data may be treated differently under the hood. There are time zone specification and handling differences. And often there's a lack of large-scale validation, so software may have bugs. Therefore, it's really important to pick a software that is consistently used for the same study, that has a source code that could be infected, and ideally that had some level of validation. Next slide, please. To just highlight again that the software choice matters, this is a letter that we published in Diabetes Technology and Therapeutics highlighting caution when using the software for the episode specifically. So the clinical definition of a glycemic episode requires the glycemic values to be within a certain range for a consecutive period of time, and typically the minimum of 15 minutes. This makes it significantly harder than assigning individual values because consecutive means you have to take into account missing data. And if you consider different levels, whether it's different levels of hyperglycemia or different levels of hypoglycemia, you have to make a choice on how do you interpret these levels, how do you impute missing data, how do you terminate an episode. We have provided the specific recommendations in this letter on what we think is the best way of handling it. And again, the software that you choose will determine how it is handled by that software. Next slide, please. In summary, I've covered 3 topics in my presentation, and the recommendations are that the 14-day of monitoring may miss long-term exposure and underpower the studies. Our recommendation is to use at least 30 days or plan for larger N, and we provide those computations in the paper. Missing data has to be specified per the guidelines because the reasons for missingness affect what is the best approach to handle it, and the choice of the endpoints may also affect what is the best approach to handle it. And there is a wild west of software algorithms and processing. I'm being asked to review new CGM software all the time that keeps coming up. So using one software that has transparent processing, that has the source that can be expected and reproduced, is really important for rigorous science. Thank you.
Thank you all for those presentations. Now I'd like to introduce Mahtab Nayati, Acting Associate Director for Therapeutic Review of DDLO, who will be joining as a panelist for the discussion portion of this session. Mahtab, I'll hand it over to you for your opening remarks.
Yeah, thank you very much. This is Mahtab. I'm the Acting Associate Director for Therapeutic Review at Division of Diabetes, Lipid Disorders, and Obesity. Thank you for this opportunity to talk a little bit about, give some remarks about CGM use. At the FDA, we do recognize the growing use of CGM in clinical practice. We understand the interest in incorporating CGM into clinical trials for characterization of various aspects of glycemic control. Historically, the technical performance concerns were real barriers to CGM adoption in clinical trials. That landscape is changing and technology is improving. It continues to improve. And I think that our regulatory thinking should be evolving alongside with it. Various CGM-based metrics have been proposed as clinical trial endpoints. Time and range is one of the most prominent ones. It's a biomarker that has not been established as a surrogate for clinical outcomes at this time, and has not been used as a primary endpoint to date for a glycemic control indication, as A1C remains the valid surrogate endpoint for microvascular risk reduction. That said, FDA does consider relevant metrics such as TIR in the clinical study section 14 of labeling, for drugs approved for a glycemic control indication where efficacy is demonstrated by change in A1C. And provided that the performance characteristics of CGM devices, data collection, analysis, everything that you heard from our speakers today are adequate. And we believe that there is clinical relevance TIR to prescribers and patients, and it could be appropriately presented when it's captured in adequate well-controlled trials in labeling. Obviously, it's a review issue, and but given that it could be a supportive information, useful information included in section 14, we evaluate this on a case-by-case basis, and sponsors should be speaking to FDA early in their drug development program, so that early engagement is very encouraged. It's encouraged to have discussions about how they want to implement CGM in their trials and what the purpose would be. So I will stop here and I'll hand it back to you.
Great. Thank you so much, Mahtab. Now I will ask all of the participants to come on screen and start our panel discussion. As a reminder, the audience can submit questions through the Q&A function. So we'll go ahead and start with a general question about what information about CGM device performance is most helpful for patients, regulators, and stakeholders using it as a DHT in practice and clinical trials? And also, what additional information would you want sponsors and regulators to make available to the public so that the trial results feel meaningful and accessible to the people living with diabetes?
Yeah, I think this is Ido from CRH. I think I talked a lot about what information we did require sponsors to collect in clinical trial and to present in our public decision-making document, as well as in labeling. Of course, we focus a lot on point accuracy because that's kind of easily quantifiable. We look at the point accuracy in various glucose ranges, like 40 to 54, 54 to 69, 70 to 180, 180 to 250, and 250 above 250. So those are the ranges that we believe are clinically relevant, and we look at that in a very granular way. And we have very granular special controls for point accuracy. For trend accuracy, we present a lot of information, but as I said, we control for the extreme discordant. So I think it's more of an opportunity for us to hear that whether these are useful and whether our CDER colleagues or the community has other kind of wish lists that we want us to consider.
I can add to that if there's time. I think that for patients, regulators, stakeholders, there are some actionable CGM performance which centers on standardized metrics. These standardized metrics are also described by international consensus. We have time and range, time below range, time above range, glucose variability. There are clinically meaningful thresholds have been described again per international consensus. For example, for TIR, and more than 5% percentage point change in time and range, according to international consensus, is considered clinically meaningful for individuals, while a 3 percentage point difference between treatment groups that could be meaningful too. These metrics are complementary to A1C. and adds additional information in terms of an overall glycemic control or hypoglycemia recognition. From regulatory perspective, as you heard throughout today, that the standardized submission protocols are needed, consistent device models throughout to avoid confounding of the algorithms, potentially separating nocturnal daytime reporting. Sleep carries some could carry a distinct hypoglycemia risk, and it's also I would also point out that I think various stakeholders could benefit from the ambulatory glucose profile. Sometimes identifying patterns helps with enabling patients and clinicians to act on whatever is needed based on the CGM information. So patterns are important. If there's any, additionally from a protocol perspective, addressing any limitations that current CGM accuracy may have, that's also imperative.
Thanks. This is great. And sort of building upon this idea of having some contextual information, we have a lot of questions in the Q&A about what other types of information may be needed. For example, whether somebody is managing their disease with medication, whether somebody has certain dietary or physical activity patterns. And then in thinking about the intended use of the CGM, what are the types of evaluations that might be needed given the intended use and potential context of use? And I think this can be relevant for any of the panelists.
I mean, from a device perspective, we look at mainly to evaluate the diagnostic accuracy of a CGM. So we look at kind of, is it accurate or not? If we compare that to clinical truth or comparative method, how close is the CGM measurement to that one? In terms of the intended use, CGM historically has been used in diabetics. And so far, with the exception of OTC CGMs or the prescription CGMs, the indication are limited to people with diabetes. But as we kind of have already done in the authorization of OTC CGMs, so with the Dexcom Stelo and Abelingo, the devices, those OTC CGMs are indicated for a broader population of people not on insulin, including people who are not with diabetes. But the clinical data supporting the authorization of those OTC CGMs are actually collected from people with diabetes. So I think we kind of have already crossed the bridge there in terms of there's still kind of technically there is still kind of on-label versus off-label use situation here. But from a technical point of view, we have used data from people with diabetes to support the performance of a device indicated for a broader population.
Yeah, I can add one more thing from the drug, like for the, as you mentioned about there could be other informations using the dietary or the timing of the food consumptions, everything. So in those kind of information might be useful for the analysis, subsequent analysis. So you can always use the other data set format as SDTM, and then that can be also used in the final analysis, whether to see the impact of the any into the like a regulatory decision. But the above all, we are always asking for the pre-specification for the clinical trials. So you should think of the ahead about what kind of other information would be useful for the submitting for the drug and then the biologic products marketing applications.
Uni, that's a great point about this pre-specification and also relates to questions that came up in the Q&A. about what happens if you have to divert from the pre-specifications. For example, you have a planned treatment and the patient doesn't come in for the treatment at the right time. Can you still leverage the data, and what are the circumstances in which you may or may not still be able to use the CGM information if it diverges from your pre-specifications?
So we totally understand about in the clinical trial, it's sometimes really hard to get into the right time for the visit. But so as long as you have collected the data, then probably you would submit to us for the additional sensitivity analysis. But we want to stick to the pre-specification of from the statistical analysis plan to use for all the data for the reliable results.
Great, thanks.
Oh,
Go ahead.
sorry. Yeah, I just wanted to add to that because I think one of those questions referred to me talking about the trial duration and window. And I think it's important to distinguish what is the total target time window that will be acceptable, and then how much days of data do you need within that window. So let's say we're talking about the second treatment visit that should be happening, let's say, in 14 days, but instead it happened in 30 days. So we should know in advance what's the length that's acceptable. Like if it happened a year later, that clearly would not be acceptable. But maybe it's acceptable in a month if we know that what we want to capture is 90 days from this period, and we need at least 30 days of 90 days. So I think there has to be an understanding of what's like the maximal period or what's the total target period, and then how much we need within that target period. Because if the duration gets really different, like if we see 7 days here, but we see 1 month over here, it gets really hard to compare.
Great, thanks. And I think relevant also to practicality, there's a question in the Q&A about participants who are blinded on CGM wanting to sort of have unblinded version. And so this question of, is there a way to compliantly unblind live CGM data in a clinical trial so patients don't need to wear two CGM sensors, one blinded for the study and the other unblinded for the standard of care? There's a mention that participants have been complaining about that burden.
I can start with that and others can add, but at the level of the protocol design, we really encourage sponsors to discuss how they want to implement CGM in their trials. And we recognize the burden of two CGMs, but it really we also are open to reviewing various proposals on how they want to implement CGM. So if you have a control arm and you have CGM randomized to both arms, any biases should be taken care of. But again, this is a discussion based on the drug development, based on the specifics of the drug development program and the utility of CGM for that particular drug development program. We're open to various proposals of how to approach it to decrease the burden as well.
Great, thanks. And we've heard about some analytical approaches, particularly from Irina and Yoon-hee. And so this question is more about the approaches for missing data, device error, sensor replacement, reporting, and what level of detail in those reporting should be expected for academic research, clinical study reports. Should that level of reporting be the same? Any other sort of recommendations?
I can start for the clinical trials. For CGM data from clinical trials is mainly aimed at evaluating treatment efficacy and safety within a trial. So for the regulatory decision on benefit risk of products, we need reliable results using correctly derived endpoints from the raw readings and appropriate statistical analysis to evaluate the treatment effects compared to the comparators. So we also want to provide the best estimates of treatment effects using CGM-derived endpoints after incorporating missing data assumptions for public information in the labeling if it is approved. So as I mentioned in my talk, the high volume and multi-layered structure of CGM data with missing data at every layer makes analysis challenging. So I want to highlight that the distinction between the no data collected and then the data collected but flagged is critical for assessing whether missingness is balanced across the treatment arms and patients. So no data collected could have relevance to treatment effect evaluation, as Irina pointed out, meaning we can't rule out its impact on the data integrity. And the data that was collected but flagged as missing with reasons can be useful for further investigation into how the missingness impacts the treatment effect evaluations. So because the ultimate goal is to evaluate the treatment effect of the product, we can start with a simple basic comparison using the proportion of missing data and the reasons for missingness, including device errors and sensor replacement, like the number of sensors across treatment arms and patients, to see whether there's any imbalance across those factors to impact to the treatment effect evaluation. So for dealing with missing data at every step, as I briefly touched on during my talk, I can bring up our recent publications on a 4-step missing data management framework, a methodological approach that progressively imputes missing data from epoch level to the endpoint. So we don't endorse any one specific method, but the main idea is how do you handle small gaps? How do you select valid days to ensure greater completeness and better coverage of 24-hour period? And how do you choose qualified aggregated endpoints for the final analysis after handling subject level missing data? So in line with our current thinking, these frameworks should be consistent with the regulatory expectation of transparency and reproducibility when evaluating treatment effects using data from clinical trials. So I hope these address the questions.
Fantastic.
Maybe add just minor comments. So as an academic statistician, I'm not representing FDA, but I think it's important to report the reasons for missingness because those reasons may indicate whether it's informative or non-informative. And that, of course, will indicate how much of an effect it may have on our final conclusions. And as I mentioned in my talk, there are multiple different reasons, and they were not exhaustive for why the data could be missing. And what I personally would do with the data will depend on what are the reasons and how much is missing to begin with.
Great, thank you. Thanks very much to our session panelists for a great discussion. It sounds like Jeff Siegel will rejoin to tell us something about new development on iStand and DHT at FDA, and then that will wrap up our session. So I will give a second for Jeff to hop on and hand it over to him.
Thank you very much, Jesslyn. For those who weren't in the early panel, my name is Jeffrey Siegel, Office Director for the Office of Drug Evaluation Sciences at FDA. At the earlier panel, there was an important development I wanted to share, and there wasn't time in the panel to do that. So I appreciate the organizers for giving me the opportunity now. So this has to do with how digitally derived endpoint developers and pharmaceutical sponsors can get input from the FDA about acceptability of their tool for use in drug development. As we talked about, pharmaceutical company sponsors can submit the proposal directly to their IND and get feedback from the FDA on that. That's a non-public process, and the broader community won't be aware of that unless somehow it's shared with them. The other route to getting feedback is the qualification program. And an instrument, when qualified, can be used in any drug development program for the particular context of use that it was qualified for. So digitally derived endpoints could be submitted for qualification in the clinical outcome assessment program. In that situation, they would be assessed based on the criteria described in the patient-focused drug development guidance series. And you can consult those 4 guidances online. Clinical outcome assessment qualifications are thorough, and they often take a long time and have typically taken quite a few years before qualification. Currently, we're adopting an alternative approach where selected drug development tools, digitally derived endpoints, can be submitted to the iSTAND program. So this is a qualification program that accepts drug development tools that are neither clinical outcome assessments nor biomarkers. And it stands for Innovative Science and Technology to Advance New Drugs. The iSTAND route may be appropriate for digital health technologies for relatively simple concepts, things like pain, cough, nocturnal scratch, where it's widely accepted that these concepts are meaningful to patients and where the outputs are readily interpretable in the setting of a clinical trial. In the iSTAND route, we don't qualify an outcome. Instead, we qualify a tool, digital health technology, for example, that measures a specific concept of interest in a specific disease or condition based on data indicating that the tool accurately and reliably measures what it purports to measure. In the clinical outcome assessment program, there are a number of other aspects that are measured, but in the iSTAND program, we would be limiting our qualification to that. We've seen the first acceptance of a digitally derived endpoint into iSTAND as a letter of intent. This was measurement of foot drop for a form of Charcot-Marie-Tooth disease, a rare condition. And we hope that this route will be used more and can facilitate assessment and acceptance of select digitally derived endpoints. Thank you very much.
Hello, everyone, and welcome back from our lunch break. I'll be moderating this 3rd session, which will cover statistical and measurement considerations in developing digitally derived endpoints for regulatory review. My name is Rachel Hendricks-Stirrup, and I'm the Research Director of Real-World Evidence at the Duke-Margolis Institute for Health Policy. And quickly, I'll share with you all that this section will explore how rigorous scientific approaches acknowledge, quantify, and communicate uncertainty throughout the measurement and review process. So by making statistical uncertainty explicit and manageable, these methods build the evidentiary foundation required for regulatory decision-making. In this context, digitally derived endpoints must adhere to the same patient-centered standards as traditional clinical measures, supported by 2 fundamental pillars, analytical and clinical validation. Next, I'll move towards introducing our presenters for this session. First, we'll hear from Vadim Zibinnikov, Professor of Biostatistics at the Johns Hopkins School of Public Health. Pardon me. Then we'll hear from Ryan Jung, a Senior Statistical Reviewer for Oncology Products in the Office of Biostatistics at the FDA. Lastly, we'll have a presentation by Lily Gerard, Acting Supervisory Mathematical Statistician within the Office of Biostatistics at FDA. Vadim, I'll go ahead and pass it over to you to get us started.
Thank you, Rachel. I hope everyone had a great break and recharged themselves. I definitely feel elevated level of glucose in my blood, so hopefully we have energy for the afternoon sessions. I'll start. So in my talk, what I'm planning to do, I plan to focus on statistical considerations behind transforming raw accelerometry data in a digitally derived measures. Can we have next slide? Yes, so some disclosures beyond academic and clinical research. I work with industry, regulators, and wellness space, but all the views are my own. Can we have next slide? Yeah, I would like to start with this picture, which dates back from 2018, which essentially envisioned that all clinical trials by now would have digital biomarkers. And although we're getting there, we're not there yet, right? And I think one of the reasons is that to derive digital biomarker, which would meet regulatory standards, there are many steps and statistical and non-statistical steps. And this is why making it regulatory level is very hard. Can we have next slide, please? Yeah, so we heard in the morning sessions about interrelated statistical considerations, and I'm listing some of them. So we definitely need to think in terms of how long the study will follow study participants to get a reliable measure. We need to think about different type of biases, which comes from devices, algorithm, different populations we work with, behaviors of those populations. Data is hierarchical, so there is epochs, days, assessment periods, which typically nested within subjects. And we need to think about how to deal with this hierarchy beyond just aggregating the data. Missingness could happen across multiple levels. Endpoint construction, so the data is very rich, and we could aggregate in averages, in variability measures. We could think about episodes of interest, quantify those episodes in terms of duration, frequency, etc. But also we could create a very novel endpoints which go beyond these traditional summaries, right? And those endpoints might be, reflective, and patients could connect to those points, but sometimes those points might be actually quite informative and might be very powerful to detect early changes, but for the patients, they might not necessarily relate that. And I think my personal perspective, which might deviate from the FDA perspective, is that we also need to think in parallel about the biomarker trajectory where, we could derive something powerful, maybe using AI approaches, but also maybe educate patients in parallel so they could understand and appreciate those early signals. And I think that these issues are also interconnected. So whatever the choices we make in one of them might propagate to the next one. So it's really important to think about this as a package. Can we have next slide? Yeah, and in terms of accelerometry pipeline, it typically has not only statistical decisions and statistical steps, but also there are many logistical steps. There are many signal processing steps. So we need to choose device. We need to decide body placement, where we're going to place it. We have to think about epoch level segmentation, what summary will represent epoch level activity. We need to detect where, non-where, define valid day, define the key activities such as sleep wake. Within wake, we need to define type of activities, maybe posture, maybe handle missing data. So only at the end of this pipeline, we arrive to the endpoint, right? And if you bring two different teams and provide this pipeline, they might use it and execute it very differently. So you might result in very different results. Can we have next slide? So this is why we need to think in terms of data provenance. So when we hand the results of a clinical trial to the statistical reviewers, they need to be able to audit the steps we did. And again, not only statistical steps, but steps in terms of choosing the right device, initializing this, dealing with missingness, etc. And they need to audit those decisions as well as be able to reproduce analytical results, right? So this is the way I would envision how the data provenance would work. Can we have next slide, please? And switching gears a little bit, in the next few slides, I'm going to talk about a regulatory science demonstration project, which was done in collaboration with our colleagues from Office of Biostatistics. And this project was co-led with Dr. Niyogi, currently at the University of Mississippi. And we focus primarily on using novel approaches for handling missingness and fingerprinting in DHT data, specifically in accelerometry. Can we have next slide? And if you think about accelerometry, of course, there are technical issues which could cause non-wear. But one of the primary driver of non-wear is essentially the person takes it off, right? And this is why non-wear, and identifying this non-wear and dealing with this non-wear is pretty critical. And missing this could be informative and non-informative, as we discussed in the morning briefly. So specifically what we focused on, trying to inform imputation algorithm, we focus on the time of the day. So can we have next slide? Yeah, and why time of the day is important. If you look at the behavior such as eating and exercise, it's highly structured within humans. So this is the data from American Time Use Survey. And if we're missing the data, say from afternoon, we definitely want to inform ourselves like what happens in afternoon, right? So we need to give more weight. So that's why taking time in consideration is pretty important. Can I have next slide? So this is where the idea of temporally local distribution come. So if you look on the left, this is a typical accelerometry gram. And we could quantify and we could represent it using this quantile envelope showing on the right-hand side, where essentially you move time window and you calculate distribution within this time window, and you understand what happens, in the morning versus in the evening. Can I have next slide? And we actually, can you go back, please? Yep. Yeah, so for example, here, if we're missing in the last day at this bottom, if we're missing this window between 6 p.m. and 12 a.m., how we could impute, how we can do epoch level imputation. We could use observed data and calculate global mean. We can look also as alternative, look at just same time window and calculate local mean within this window. And it could be very granular, maybe for every single minute. We could look at all available data and sample from that and fill it in. Or we could actually sample just from that window, which I'm showing here in red, and can condition on time. And this is the 5 different approaches we explored across multiple missing patterns in multiple simulation scenarios. Can we go to the next slide? And if you look at this panel on the left-hand side, it shows essentially difference between target distribution you could construct using all observed data minus our estimate. And ideally, you want to have it just at 0, but we see that using fixed imputation, using either global mean or local mean, you have this constant bias, and it's represented in terms of bias in specific intensities of activities. But when you use a local distribution and local neighborhood distribution, the bias is much smaller. It's still present, but it's much, much smaller. And we saw similar pattern across different results, across different simulation patterns we looked at. Can we have next slide? And in addition to conditioning on just time of the day, you could also borrow information across participants, which is similar to the participants you're working with, right? So you could expand this framework. You could also use this framework for subject level fingerprinting. So you could compare, this envelope profiles, which you can see on the bottom, right? And for example, in this case, one subject has this red pattern, and the other subject has this green pattern, and we see that morning hours and the nighttime hours have much more discriminatory power. We see much more difference. So you can actually understand whether data comes likely from the same subject or not using this framework. Can I have next slide? And just a few concluding remarks. So we've been talking about CGM in the morning, and we talk about actigraphy now and accelerometry, and the measures might look different, moderate vigorous activity or time in the hyperglycemia, but in reality, there is a lot of structural similarity from statistical perspective. I mean, we get those events by just applying the threshold, right, for accelerometry amplitude or for CGM, right? And we could model those events using exactly the same statistical methods, using durational properties, number of bouts, distribution of time between the bouts, etc. Can we go to the next slide? And just a few resources. So for those who's interested in the statistics in terms of wearable digital health technologies, we actually have mobile and wearable data science interest group within American Statistical Association. It was recently established, and you see the link on the bottom. We're also releasing a guide to wearable and digital health technologies where we review modalities, metrics, and methods, and we tailor this guide to biostatisticians and statisticians. So you're very welcome to read it. It will be available in a couple of weeks. Okay, can we go to the last slide? Yeah, and this is another great resource which coming up. It will be available in a couple of months, my understanding. So this is also a community effort. So great resource. And I think that's it. Last slide, just to tell you thank you. And I just want to mention just one concluding sentence that the digitally derived endpoint is a pipeline, right? It's a sequence of many statistical and non-statistical decisions, and this is why we need to think about this as a package. Thank you.
Thank you. Next, we'll pass it to Ryan Jung.
Thanks, Rachel, and welcome everyone. My name is Taeyeon Ryan Jung from CDER at FDA. Today, I'll be presenting on analytical validation as the foundation of fit-for-purpose digitally derived endpoints. As DHT becomes increasingly integrated into clinical trials, it is important to establish what constitutes rigorous and credible evidence for regulatory use. Next slide, please. I will organize this presentation around 3 parts: what analytical validation is and why it matters, the key evaluation components, and finally, a reviewer's perspective. Next slide, please. So first, I would like to distinguish verification from validation as described in the FDA guidance for remote data acquisition. This matters because a device can function perfectly from an engineering standpoint, but still produce measurements that are systematically biased or imprecise. Verification asks a fundamental question about the technology itself. Was the technology built correctly, and does it capture the data as intended? In other words, is the device working properly? And this confirms that the DHT performs according to its technical specification and produce raw data that accurately reflect the signals it is intended to capture. Validation, on the other hand, shifts the focus from the device to the derived output. It asks, is the measurement scientifically sound and clinically relevant? And this step confirms that what the DHT produces is not just technically captured, but scientifically meaningful and fit for its intended clinical purpose. And together, in sequence, verification and validation provides the evidentiary foundation for an output to be considered a digitally derived measure. And that measure that can then, when appropriate regulatory criteria are met, be developed into digitally derived endpoint. Next slide, please. Within validation, there are 2 distinct questions in sequence. The first asks whether the algorithm integrated into the device measures the target parameters accurately and reliably. The second asks whether those analytically validated measurements are clinically meaningful for the patients. My presentation focused on the first question, the analytical validation. And after this presentation, Dr. Jarrett will discuss the clinical validation. Analytical validation demonstrates that a digitally derived measure represents the intended signal within its intended population, setting, and conditions of use. And this requires evaluating the entire measurement pipeline and characterizing its performance against an appropriate reference standard. Importantly, a favorable analytical validation finding demonstrates adequate performance under the conditions evaluated. It does not, by itself, establish generalizability beyond those conditions, clinical meaningfulness, or fit-for-purpose status for regulatory use. Next slide, please. So why does analytical validation matter? Traditional endpoints are generally collected at discrete clinic visits under relatively standardized and controlled conditions. In contrast, digitally derived endpoints are derived from continuous, real-world measurements, uncontrolled conditions, and often exhibit pervasive structured missingness. As a result, their measurement properties cannot be simply assumed, and they need to be empirically demonstrated. Analytical validation provides that evidence by evaluating accuracy, bias, precision, and measurement error to support fit-for-purpose evidence. Without analytical validation, there is no scientific basis for determining whether the digitally derived measure reliably reflects the intended signal. During the review process, our goal is to understand how well the measure performs, where the error arises, and how performance varies across subjects and conditions. Next slide, please. A digitally derived measure is not simply a sensor reading. It is the product of a multi-stage processing pipeline. In each stage, multiple individual processing decisions can affect the quality and the interpretability of the final output. Analytical validation, therefore, needs to consider the entire measurement pipeline, not just the final output. It is important because Errors introduced early in the pipeline can propagate forward and ultimately compromise the validity of the final measure. Next slide, please. A reference standard provides the ground truth against which a digitally derived measure is evaluated. And choosing that reference standard is a scientific judgment, not simply a procedural step. Any claim of validity is only as good as the reference used to test it. Therefore, the reference standard directly shapes the strength of the validation conclusion. Because there is no universal gold standard, the appropriate reference depends on the context of use. Also, every reference standard has its limitations, and those limitations need to be considered when interpreting the results. And when the reference standard itself relies on human judgment, such as manual venue annotation, its own reliability must be also quantified. If the annotators do not show consistent agreement, the reference itself may not provide a credible ground truth. Next slide, please. One useful way to organize the major sources of measurement error is to organize them into three broad categories. One important source is measurement distortion, which can arise from the device itself and from the algorithm. We also need to consider data loss that can result from non-wear, device removal, missing data, and dropout. And importantly, these patterns of missingness may be systematic rather than random. And finally, we need to think about generalization, where performance may vary across measurement setting, population, or disease states. For example, a measure that performs well in a controlled clinical setting may behave differently in a home environment. Well, these are not rigid categories, but rather a practical framework for organizing the major sources of error that may need to be evaluated during the review process. Next slide, please. The choice of performance metrics should align with the type of the output the algorithm produces. For algorithms that make discrete classifications, such as scratch versus no scratch, we can look at the metrics such as sensitivity and precision, for example. For continuous outputs, such as total sleep time, we may instead focus on measures such as bias and limits of agreement, for example. In both cases, performance is evaluated relative to a reference standard, and the appropriate metrics depends on the type of the output and the context of use, and often, no single metric tells the whole story. And that is why choosing metrics that match the algorithm output is important. And using a metric that doesn't fit or rely on a single metric alone can give a misleading picture of the performance. Sponsors should therefore pre-specify and report a set of performance metrics appropriate for the algorithm output and its context of use. Next slide, please. One of the most common and important errors is the conflation of correlation with agreement. These are not the same thing, and they do not answer the same question. Correlation tells us whether the values move together. Agreements tells us how closely the values actually match. A high correlation can look reassuring, but we should be careful not to overinterpret it. High correlation does not confirm that the digitally derived measures are numerically close to the reference. There can still be substantial systematic bias. So the key point is simple. Correlation alone is not sufficient evidence of agreement, and agreement should be also assessed with appropriate methods. Next slide, please. Once we have the performance results, the next question is whether they are acceptable. That requires a pre-specified criterion, and choosing that threshold is itself a scientific judgment. The threshold should be set in advance and justified for the intended context of use. We also need to evaluate acceptability for individual sources of error rather than relying on an overall performance. An average can look acceptable while still hiding an important problem in a particular setting. And finally, acceptability is context-specific. What is acceptable in one context of use may not be acceptable in another. Thus, the overall analytical validation conclusion is built from these individual source-specific determinations. Next slide, please. From a reviewer standpoint, we often see some of these common mistakes shown here. Some better practice include validating the right population, looking beyond the correlation, and clearly documenting the software and algorithm version being evaluated. Well, these may seem like basic points, but they are important for ensuring that the evidence is relevant, interpretable, and reproducible. Next slide, please. Before I finish, let me highlight a few key points to keep in mind. First, analytical validation is inherently context-dependent. There is no single approach that works for every measure. What is appropriate depends on the intended use. Second, every stage of the measurement pipeline is a legitimate and necessary part of validation, not just the final output. Each stage can introduce error, so each needs proper review. And ultimately, the goal is not perfection. The goal is to understand the measurement error well enough to determine whether the measure can reliably support the question being asked. And finally, I would encourage early engagement with FDA because early discussion can help clarify expectation, identify potential issues, and support a more rigorous and efficient validation program. With that, I hope these considerations provide a useful framework for thinking about analytical validation. Thank you.
Thank you, Ryan. Lastly, we'll turn it to Lily Gerard.
Thank you, Rachel. Hello, everyone. I will discuss developing patient-focused digitally derived endpoints from a quantitative measurement perspective. Next slide, please. So before getting into the specifics, let's take a look at a high level at how the data flows from digital health technology to the digitally derived endpoint. Here's an overly simplified example. In practice, each step would be far more precisely defined. The purpose of this slide is to illustrate the conceptual pathway, not to prescribe the metrics. Starting with an important assumption, and this is really our starting point. Based on qualitative evidence, adult patients with disease A want to see improvement in walking, specifically ability to take continuous steps. And medical product XYZ has the potential to improve taking steps based on the proposed mechanism of action. With this knowledge, we decide that a wrist-worn triaxial accelerometer as the DHT could be the appropriate device to use. At this stage, we need to conduct verification of the DHT. In other words, for adults with disease A who wear the wrist accelerometer continuously, does the hardware accurately capture the correct signal? This is followed by analytical validation, answering the question of whether the device and the algorithm accurately detect stepping versus a reference standard. For example, we may consider validating the step detection algorithm against a video-annotated ground truth where a trained rater manually counts steps. After the raw sensor data is processed through the algorithm, many stepping-related metrics can be generated. Which one do we choose as the digitally derived measure? This is where qualitative evidence from patients again drives the selection. Earlier, qualitative evidence told us that ability to take continuous steps matters to patients. Now it tells us which aspects of stepping matter most. For example, we identified that step count and cadence of longest continuous stepping periods are important and meaningful to patients. At this point, clinical validation becomes relevant. Is the selected target metric, examples, step count, considered fit for purpose? How should it be operationalized? And next, we construct digitally derived endpoint based on the digitally derived measure. An example endpoint may be changed from baseline in weekly average step count at week 24. Clinical validation comes to play again, helping us understand whether the endpoint reflects an aspect of the patient's health that is meaningful and whether it is capable of detecting treatment effect within the context of the planned clinical trial. This is also where we establish its clinical meaningfulness. All of this is basically saying you need to be able to use the endpoint in the statistical analysis of the clinical trial and make sense of everything in the end. Next slide, please. To better understand the endpoint development process, it is useful to introduce the patient-focused drug development, or PFDD, framework. The PFDD process begins with understanding the disease and identifying what is meaningful to patients. This leads to identification of the meaningful aspect of health, the concept of interest, and the context of use. Only after these concepts are established, we move to selecting, developing, or modifying a clinical outcome assessment, or a COA, and evaluating whether it is fit for purpose. A COA is a measure that describes or reflects how a patient feels, functions, or survives. For example, a patient-reported outcome. Finally, the outcome assessment is incorporated into an endpoint. Next slide, please. Digitally derived endpoints follow the same framework. We still begin with understanding the disease, identifying a meaningful aspect of health, a concept of interest, and a context of use. The main difference is that now we need to determine whether the outcome can be measured appropriately using a DHT while selecting the digitally derived measure. We also need to evaluate the fitness for purpose for both the DHT and the resulting digitally derived measure. So while the technology may be new, the underlying measurement framework remains the same. Next. And next, we will take a closer look at these two steps in the red box. Next slide, please. This brings us to clinical validation specifically. Clinical validation provides evidence that an analytically valid digitally derived measure appropriately assesses its intended concept of interest and corresponds to a meaningful aspect of health. It helps demonstrate that differences or changes in a digitally derived measure reflect meaningful differences or changes in how patients feel, function, or survive. This concept will be central to the remainder of the presentation. Next slide, please. The technology, or the DHT, should not drive the selection of the digitally derived measure. Instead, the digitally derived measure or the outcome measure should be driven by the concept of interest identified from talking to patients and/or caregivers. The key questions are, is the outcome measurable by a DHT within the context of use? And is it appropriate to measure that outcome using a DHT? These questions should be answered from a patient-focused perspective. Next slide, please. Now we evaluate fitness for purpose. Importantly, the fitness for purpose should be considered for both the DHT and the digitally derived measure. The PFDD framework provides a useful structure for the evaluation of fit for purpose, which is where I will turn next. Next slide, please. PFDD Guidance 3 presents an evidence-based rationale framework and describes 8 generic components that should be considered for supporting evidence that a COA is appropriate given a context of use. These principles also apply to DHT and digitally derived measure. You should consider whether all components and subcomponents are relevant to the use of DHT, and if there are specific considerations unique to the DHT that need to be taken into account. Next, I will use component E as an example to walk through some considerations relevant for clinical validation. Next slide, please. Component E concerns whether the scoring for the digitally derived measure is appropriate for assessing the concept of interest. Using stepping as an example, we can see there are different levels of scoring. We begin with raw sensor signals. These signals are processed into short-duration observations, such as epochs or events. Those observations are then summarized into a daily summary metric, for example, total daily steps. Finally, those daily metrics are aggregated over an assessment period, such as 7 days, to produce a score which can be used to construct a digitally derived endpoint. Next slide. An important scoring consideration is missing data. It is important to understand both the amount of missing data we're dealing with and the reasons behind it, whether from device malfunction, transfer errors, compliance issues, or patient's health status that lead to non-wear during the morning monitoring period. Once that is established, we need a clear definition of what counts as missing data, methods for handling it, and careful checks on assumptions, including sensitivity analysis. A key question is how much data is enough to produce reliable estimates of digitally derived measures. Within that, here are some example questions to ask. What is considered a valid day? I put in quotes here because this is a term we often see in submissions. How should we determine minimum wear time within a day? What constitutes appropriate assessment periods during the day? Are we looking at 24-hour monitoring? Or are there specific time windows that are most clinically relevant for our endpoint? What is the minimum number of valid days required within an assessment window? Next slide, please. Now on to developing digitally derived endpoint. It is similar to developing other clinical endpoints. A clinical endpoint should be a clinically meaningful reflection of how a patient feels, functions, or survives. A well-justified rationale for the choice of endpoint should be provided early in the planning of a clinical trial. These guiding principles are not new or unique to endpoints based on data collected from DHTs. Next slide, please. Again, borrowing the guiding principles from PFDD Draft Guidance 4, a well-justified rationale to support a chosen digitally derived endpoint will need information on the following. In the interest of time, I will not read through all. The bottom line is that having a well-justified rationale not only helps sponsors facilitate clear communication with FDA, but also helps facilitate more efficient review and decision-making by FDA. Next slide, please. So throughout my presentation, I intend to show that the principles described in the PFDD guidance series can be applied to the development and evaluation of digitally derived endpoints. This always starts with incorporating input from multiple stakeholders, especially input from patients and our caregivers. Clinical validation is critical. The requirement of fit for purpose applies to both the DHT and the digitally derived measure. Thank you.
Thank you, Lily. And thank you to everyone for the informative presentation. I think I hear an echo. Okay. Thanks again, everyone. Now I'd like to introduce the panelists who will be joining us for the next discussion portion of this session. First, we'll have Jens Prosgaard, Director and Data and Statistical Strategy Leader in Digital Health at Sanofi. Then we will also have Mitchell Soka, Senior Physician in the Division of Nephrology in the Office of New Drugs within CDER at FDA. So, Jens, I'll turn it over to you to get us started with your opening remarks.
Well, thank you, Rachel. We've heard about statistical analysis perspectives and evidence generation from an academic and regulatory side here, and I'm glad and honored to be asked to give some sponsor feedback on this. As sponsors, we very much depend on actionable information and purpose-driven analyses throughout the stages of endpoint development, from to a proof of concept through later clinical validation and even profiling drugs already on the market. If you, for instance, want to see if you can get a positive signal on cough monitoring for a drug that's already on the market for COPD. So the header for my remarks to the three talks will really be the sponsor need for disciplined and actionable analyses. And so towards that, Dr. Ziponikov expands on statistical issues, in particular missing data and rules from imputation from an academic point of view. Some ideas from a tri-list point of view that come to mind are these, that we as tri-lists are not necessarily interested in individual accelerometry trajectories. The end game is to detect a mean group difference between two arms of a clinical trial. And from this perspective, one should ask about the purpose of imputation for missing periods for individual subjects. So is this something, imputation is normally helpful if it enables us to use standard software to fit a model with incomplete data, so as not to not to discard data, but the purpose of why we impute and why that's helpful for the analysis should be made up in advance. You can take a cardiopulmonary population for an example. If we use time spent in moderate to vigorous activity as an endpoint, normally there are rules which discard patient days on data which is certain where time is not met. Should the missing activity classification be imputed here? Again, it depends on the model. If you use a simple Markov chain approach, you can argue that the subject spends time in various activity states and transits out according to specific transition intensities. And such a model doesn't throw individual days out, but it leads to an analysis with a wear time weighted error variability with really no need for throwing anything out, and the missing data problem vanishes. In the interest of time, go over to what I heard from the take home from Dr. Young's talk is a nice description of what a reviewer looks like in assessing analytical claims made by a sponsor. And there are some conundrums that we're all in agreement about here, that purported gold standard is not often a gold standard, and the warning against relying solely on vague claims of correlation and the need to really do validation studies in the intended trial population. And to me, this is just as important for regulators as for sponsors who, before we come to the confirmatory stage, need to generate information for actionable internal decision making. Do we, in a small proof of mechanism or proof of concept study, trust these measures and devices so that we want to make the big investment in putting it into a largest 2B or 3 trial? So this goes back to the idea of actionable information. Statistical analyses of analytical data should have a clear purpose. Boundaries for acceptable system performance should be set up front, and this by necessity has to do with a view to the intended application in a clinical program. For example, an analysis of gait speed by accelerometer done and done in a gait lab by video capture, it might result in a report of an R-square between two assessments and a report of an interclass correlation for measurements taken at different days and a Bland-Altman plot for agreement. But without a clear pre-specified notion of what claims, this often doesn't become an actionable analysis, but just a record of a certain activity that was done with data. And this is really what I recommend and look for in a sponsor. Don't just do the analyses you can do what's actionable. Now, fortunately, the statistical challenges in analytical validation are not new. Many of the problems have been solved in the chemometric and assay validation literature going back a very long time. In particular, the problem of comparing two methods when none is a gold standard. It's been known since really the foundation of our science that a simple linear regression slope is biased downwards when there's random error in the purported gold standards. So I try to and would recommend to other sponsors to familiarize themselves with latent variable modeling, errors in regression techniques, Simex estimation, and even a very basic level, often a simple Bland-Altman plot, as the one shown in Dr. Young's talk, may hide two methods which are well suited, even though at first interpretation one would say, yes, they're outside the limit here. This is not a good second method. And again, it goes back to the point again, the point is not necessarily more or more technically sophisticated analyses, but more discipline in analysis. Now, Dr. Gerard very pointedly focuses on clinical validation in the context of focused drug development. That's a key interest to any sponsor. Sensor-based endpoint hold the promise of measuring novel COAs, capturing patient disease in a way clinical endpoint may not, sleep quality and irritable bowel disease, tremor in kidney transplant, scratch in atopic dermatitis, novel endpoints that are related to what patients say is important to them, and more so than the standard clinical endpoints. Real quick here, if I can, to Dr. Girard's argument about how should an argument be made. Well, she goes into the very important notions of anchor analyses against established measures of patient impression of disease and what is outlined in the draft guidance 4 of the patient-focused drug development series. I would encourage every sponsor to read that very carefully and contribute to that, and gaining experience with COA changes and COA analysis. And in particular, after a point, it might be possible to phrase those recommendations in a predictive framework. In other words, if you see a certain change in your novel COA, is that predictive of a certain change in a patient measure of satisfaction and so on? So I would again, encourage the sponsor to get familiar with ordinal regression techniques, get familiar with the whole item response analysis framework, and be able to contribute to that particular these particular ideas and developing models for COA validation.
Yeah,
and I think I'll stop here. Sorry if I went too far.
No, no worries. Actually, we'll turn it next to Mitchell to provide our final opening remarks before we move into our general discussion.
Thank you. Thank you for the opportunity to be here. Again, I'm a cardiologist in the Division of Cardiology and Nephrology, and we've seen a number of these discussions come through our division. And so I'll speak a little bit from experience, but I think it is, I want to make a couple points. One is all of the variabilities of measurement and assessment that have been talked about already today are critically important for getting to what is all of our intention, a clinically interpretable endpoint. But that's where I want to really focus. And I want to make the point that there are a lot of ways to have a digital derived measure from a DHT that may not be the creation of a novel endpoint. So you can imagine digitizing and using a digital health technology to assess a 6-minute walk test distance, which is a known measure that we use to assess functional status. And you're just trying to validate the novel measurement to align with what is an understood clinically validated and clinically useful and interpretable test. In the setting of actigraphy, we have a novel measure that the validation of the measurement of the digitally derived measure is unclear. It's unclear how to both what aspects of the actigraphy measurement are most aligned, both correlated and actually accurate on a sort of Land-Altman perspective. And then most importantly, how to interpret any clinically meaningful change. And in the end, when you're trying to use a digitally derived measure to demonstrate a beneficial clinical treatment effect on how a patient feels or functions or survives, you need to be able to know that the change that you're seeing on the measure is clinically meaningful to patients. And this is often where we see that there is more work to be done to really end up in a place where you can interpret the end result of what you're measuring. And I think that if we just look back historically, conceptually in the space, particularly of actigraphy, it always sort of made sense on face value. Well, if we measure everybody more, what they're doing out in their day-to-day activities, if we are just able to assess in kind of real life what is happening, that we will have a better sense, that it will more easily translate into understanding that patients are better with a therapy or not. And I think what has, as we've, I think, gained some wisdom in this space and others, we've realized that there still is a lot of understanding that is missing in terms of interpreting all of the data that can be collected to understand what is which concepts are best represented by actigraphy measurements and which ones can be measured within the realm of measurement error and variability of measurement, and then which of those can potentially be used to interpret a clinically meaningful benefit with an intervention. And so I just wanted to raise those concepts, which I think are critical in the interpretability end. And thanks for letting me make some comments.
Of course. Thank you so much to everyone. So before we move to the discussion portion, I'm going to briefly recap the evaluative framework presented today. So Vadim, you first provided an overview of statistical approaches to addressing missing data in DHT studies, focusing on missingness at the epoch, day, subject, and endpoint levels. And then we heard that establishing the validity of a digitally derived measure, that rests on two distinct but complementary pillars. The first is analytical validation, which asks whether the measure accurately and reliably captures the intended signal within its intended population, setting, and conditions of use. The second is clinical validation, which asks whether that measure is clinically meaningful by reflecting on how patients, again, feel, function, or survive. These two forms of validation answer different questions, essentially, yet both are essential. Neither on its own is sufficient to establish confidence in a digitally derived measure. And with that said, I'm going to move into our group discussion. As a reminder, the audience can submit questions through the Q&A function through Zoom. So as we get questions from the audience, I'm going to ask my first question really for Vadim and Ryan. So what does measurement involve for a digital endpoint beyond the sensor itself?
Yeah, maybe I could start. So as I described in my talk just a little bit, that it's not just data coming from the sensor. There are like a sequence and pipeline of decisions, and many decisions not statistical. And I think, down the road, you can do many different choices. And this is where I think it's incredibly important to think about community level and industry level harmonization efforts, right? Like we can learn a lot from data intensive fields where there is one big study, 20 different teams try to process it, get 20 different results, discuss why it's different, right? And converge to something. And I think, again, this is why I don't know, maybe working in the pre-competitive space, using resources with Digital Medicine Society, or it might be part of PDUFA, but this type of efforts I think will be important to really move the field forward.
Sure. And I think that Vadim gave a very important and practical insight, and I want to build on it from my regulatory perspective. So a high quality sensor alone doesn't guarantee a valid or meaningful measurement. The sensor is only one component of the measurement system. So, and the raw signal still has to pass through multiple processing models, including like signal pre-processing, context detection, segmentation, until it become a digitally derived measure. And each of these stages require important analytical decisions, such as what level should be treated as noise, or whether the device was actually worn or not, and where the event begins or ends. And because measurement is produced across this entire pipeline, the measurement error can arise at any of these stages. And the important question is whether the sources and the magnitude of that error have been adequately characterized and are acceptable for the intended use. And this is why each relevant stage of the measurement process can be a legitimate target of validation. So looking at the relevant parts of the measurement process beyond the sensor itself can help identify where the errors are introduced.
Mm-hmm. Yeah, thank you so much. Another question. The fit for purpose framework applies to both the DHT and the digitally derived measure. So what are the most critical considerations when evaluating whether a digitally derived endpoint is fit for its intended regulatory use? Lily, Mitchell, would love to hear from you both on that question.
Thank you, Rachel. I think, Mitchell, I'll get started and then you can add more to it. So in my presentation, I spent some time talking about fit for purpose, and I highlighted the 8 components comprising this evidence-based rationale from the PFDD Guidance 3 that can be adapted for evaluating fitness for purpose of digitally derived measures. So I just want to emphasize that the 8 components are not intended to be used as a checklist in any way. These 8 components are not always the components that matter in your situation. At times, some of them may not apply, or other times, other components or subcomponents will be needed. So you really need to think about, your particular program. So it should be used as a starting point to help us all think through what is considered relevant to help build that evidence for fit for purpose, right? From my perspective, it's not about what analysis we need to conduct. It's about why we need to do an analysis. Ryan talked about the correlation analysis. We see that often, but we need to ask, why are we doing that? What is the purpose of that analysis, and what can it tell us in the end? So I think I'll leave at that and see if Mitchell wants to add anything.
Yeah, I think when we're talking about fit for purpose, it comes down in the end to the clinical interpretability of whatever measure you're designing. And I think we have, and if I just want to go off of the lead in from question 1, which is all the sources of potential measurement, error or bias, or that all of those decisions that you make regarding your measurement, regarding your analytic framework, regarding your algorithms that you use to reach your digitally derived measure from whatever data is being collected in the raw, and then however you end up analyzing those measures, all of those decisions affect, unfortunately, the clinical interpretability of the measure. And it is very critical to realize that you can take a measure that potentially could be clinically interpretable and make it not interpretable by decisions you have made much earlier in the process, creating that kind of foundation of data collection and measurement that you're achieving. And so, assuming that you reach a point where all of those basic fundamental aspects of how you're measuring and deriving, creating your data points are valid and reliable and measuring what you say they are measuring, you then still have to have this place where the data that you are looking at represents, can be interpreted as clinically meaningful. And this gets back to what I kind of said in my lead in, which is we need to know what the relationship is between those measures and the patient experience. And that not just that those things predict maybe patients who are at higher risk of a downstream clinical event, because that is really a separate bucket than that the measures are the clinical experience and that the measures are what the patient is experiencing and that we can take them as a true clinical endpoint. And those are very different. And so in the end, when we're thinking about creating a novel digitally derived measure, a novel endpoint from these digital measures, there is no one way to achieve that, as Lily just said, but it really is going to evolve. And I hope you've heard throughout the day that early and often discussion with the agency to arrive at a validation framework that is going to demonstrate that what you're producing from your electronic or digital tool is something that we believe that you have convinced us, that you can convince the patients is something that is truly meaningful to them. And then again, that we can see an actual treatment effect. And those are quite high bars, is what we've seen historically. And I think that that's the critical component to keep in mind. It's going to take a lot of work to achieve what is a clinically interpretable endpoint.
Yes, thank you both. And I will add that another thing that has at least stuck with me, and I would assume many of you throughout the day, is that the goal is really to just understand error well enough to determine whether the measure can support the question being asked. I think that is a very salient truth and point for this discussion. So with 3 minutes left, I will ask one question, really targeted toward Ryan and Jens. And as we wrap up, I would love to know from you both. When a digitally derived endpoint relies on a machine learning or AI-based algorithm to convert raw sensor data into a clinical measure, what level of algorithm transparency and documentation does FDA expect? And is there a preference for interpretable models? For industry, and this is for Jens, how do they choose the algorithm and the strategy used to implement in study design? We've got 3 minutes left.
Yes, thank you, Rachel, for bringing up this question. And this is an important question because we are seeing increasing use of AI-based algorithms and DHT application, and I expect that trend to continue. And FDA generally doesn't require a sponsor to use a particular type of AI algorithms. The choice is essentially the sponsors, and whether it is sophisticated or state-of-art AI models or just a simple or conventional algorithm is not what matters. The key is whether there is sufficient evidence to evaluate the performance and reliability of the resulting measure in the intended context of use. So we expect the sponsors to clearly explain what their AI algorithm is intended to do within the overall measurement process, how it processed the inputs, how it was trained and referenced, and whether those data and its use are appropriate for the target population and the conditions of use. And to speak about transparency, we don't require every single aspect of the AI-based model to be directly interpretable. Rather, FDA needs enough information about the model's development, the performance, and limitations to determine whether the resulting digitally derived measure is reliable for its proposed context of use. So that'll be my answer.
Jens, I think you're talking on mute.
Let me just add to that as a sponsor, we are also aware that there still has to be content validity. That if we can come up with a black box supervised regression learning system that can make some predictions that are close to Parkinson's MDS-UPDRS scores, it still has to be credible. It still has to be explainable to stakeholders, to KOLs, and so on, what is really driving these algorithms. That it can't be totally black box. There has to be a sense for what covariates contribute here. What is the weight model weighted towards? What is the end of it? Because it's if you just do blindly fitting models, you can end up in paradoxical situations. You can end up in situations where a pain algorithm is completely driven by a proxy for disease status and not fluctuation of pain over the course where you are wanting to use the algorithm. So content validity has to be kept in mind here, irrespective of how you design your regressions.
Absolutely. Thank you all very much for being here for our session 3. Now we will turn it to our session 4 panelists and presenters.
Thank you very much. Hi, everyone. I'm Laura Lee Johnson, and I look forward to moderating our 4th session, where we'll discuss the challenges in analyzing and reviewing digitally derived endpoints and focusing on missing data, regulatory review lessons, analytical decision-making, and best practices for rigorous, consistent evaluation. I will start by introducing our presenters. We agreed to keep this short. So first, we're going to hear from Suni from Stanford University, and then from Cynthia from FDA. So Suni, I will pass this over to you to get us started.
Thank you, Laura Lee. My name is Suni Goodbole. I'm a senior biostatistician at the Quantitative Sciences Unit at Stanford University. We're a group of collaborative statisticians that are working with our medical community for different research studies. And today I'll be talking about missing data imputation for novel digital health technology-derived endpoints. Next slide. So at the QSU, we've been using a couple of different digital health technologies, including the Apple Watch, accelerometers, continuous glucose monitors, and what I'll be talking about today, which is screenomics, which is actually a study in which we captured screen data from adolescents' iPhones. And so that was a way for us to get an idea of the types of the way they were using their phone. We have to say with all of these digital health technologies, we found that there's nuance in the types of problems with missing data that we encounter and the solutions that we have for them. Next slide. So research questions for digital health technologies, I think we've covered that a lot today. For me, looking at the screnomics data, we were actually trying to relate social media engagement to daily mood. And we were trying to see, one of our first questions, of course, is can social media engagement be objectively measured by screenshots of social media app use? And while it's probably not a complete assessment of social media engagement, it does give us at least one dimension of engagement with social media. With accelerometer data, as we've seen questions about daily physical activity and relating that to sleep quality. These can both, of course, be use accelerometer data, but we need to define the temporality of the association that we're looking at. So are we looking at daily physical activity contributing or influencing sleep quality, or are we looking at a night's sleep quality influencing physical activity the next day? And then, of course, we can use digital health technologies for just-in-time interventions. The quantitative sciences unit was useful in trying to establish the validity of the Apple Watch in terms of detecting atrial fibrillation. And we've also been using this for like detecting hyper and hypoglycemic events with continuous glucose monitors. And those could be points of intervention for different research studies. Next slide. So to focus on statistical challenges, of course, one of our challenges comes from the high frequency of the data that's captured. That creates a computational challenge. And then also missingness in that data can be from things like non-wear, and that can entangle with kind of device malfunction or device syncing issues. Data are also aggregated to obtain estimates. So one of our questions, of course, is how do we aggregate the data so that the estimate is representative of the construct that we're looking for? And our short periods of time, how are they related to like a whole day of data? And can we borrow strength from other participants or multiple days across one participant? So on the right hand, I'm showing some examples of complete sensor and missing sensor data. And with each of these sensor technologies, we might use a different strategy for imputation. So up at the top, you can see there's temperature data, and we see missingness within that. We might be able to borrow information from other days or other monitors to help us impute those missing data. At the bottom, you'll see kind of a pattern of a day's temporality. And in that first day, what I've labeled day one here, we've actually seen that there's some drop off after midnight in terms of the amount of data that's collected. And so the question is, does the data that we have actually represent that whole day, or do we need to find a way to impute kind of the missing part of that day? And then in day two, since we have very little sensor data to go off of, do we consider that, do we try to impute that day, or do we knock it out completely from our analyses? Next slide. So for statisticians, the types of data, missing data mechanisms that we're interested in or that we pay attention to are informative and non-informative mechanisms. I think Vadim talked about this a little bit in his talk. The first one, of course, is missing completely at random, and that really means the data are missing without a relationship to the observed or unobserved data. So we can think of a server going down and leading to missing data after recovery. We consider this a non-informative missingness, and so it shouldn't actually affect the estimates that we're trying to get because the mechanism doesn't relate to the underlying data. And then missing at random is when data are missing systematically, but can be fully explained by other variables that are observed in the data. And we can think of this as like missingness being more likely in boys versus girls when we have that auxiliary variable of boys and girls, and we're able to actually impute data in a way that allows us to recover a valid estimate of our association. And then finally, missing not at random is kind of the most pernicious form of missingness. Here, the missing data directly depend on the unobserved missing value itself. And we can think of missing data for participants that are more sedentary during sedentary periods. This is likely going to bias our estimates, and we need to find ways to, at least from my perspective, statistically impute data to give us kind of a more reliable estimate of our effect. Next slide. So one way to handle missing data is through multiple imputation. It can be computationally challenging for high frequency data and high correlation between samples, but the tools for missing data are pretty accessible, or multiple imputation are pretty accessible. And there's kind of a wide literature on this. And so it's one of the methods that we wanted to use to try and attempt to solve a missing data problem. And so there's two types of multiple imputation. There's a passive multiple imputation in which we impute base variables and use a specified relationship for imputation of derived variables. And one way to think about this is with the example of BMI. BMI is a function of weight and height. In a passive imputation model, we would be imputing weight and height measures and then creating an imputed BMI derived variable from those base variable imputations. And then with the active imputation, we'd be actually imputing both the base and the derived variables and making sure that our model included kind of all auxiliary variables that we think are important to the missing data mechanism. Next slide. So screenomics is kind of an example of this. Again, we were looking for a relationship between social media engagement and mood. Mood was collected in a survey measure that happens at a daily level, and social media app use is collected in screenshots on phones at 5-second intervals. And so we had a process to classify these images into different app use categories, and we had 6 different kind of apps that we considered the biggest social media apps and were used to combine together to create kind of our assessment of social media app use. And then in terms of missing data, what our research team decided to do was create a data rule. And so we had an interclass correlation of 0.8, and we looked at every level of our data and made sure that the amount of data that we had for every minute, for every hour, and every day was actually within a 0.8 or above a 0.8 correlation with complete data. So we were using complete data as kind of our ground truth. And so in our next slide, please. So for the simulation, what we were trying to do is just test the data rule and to figure out if multiple imputation strategies were kind of more powerful than using the data rule. So we created 2 different data sets of 20 participants each. One was highly correlated, one was moderately correlated. We created a simulation model to derive in kind of an outcome variable for us. And then we used different missing data mechanisms to pull out specific hours of the day. We were looking at an hour to a daily level imputation. And then we were comparing kind of days at threshold and then passive and active imputation strategies. Next slide. So these are kind of results. They're still under construction. We're still tinkering with the amount of iterations we're doing for our simulations and the number of imputation data sets that we're getting. Our takeaways are that both for missing at random and missing not at random conditions, the true associations are underestimated for using just the threshold values as well as using active and passive imputation. Under the missing at random missing data mechanism, active multiple imputation performs better than using the threshold in highly and moderately correlated data, but the difference is larger for highly correlated data. And then under the missing not at random mechanism, multiple imputation and the threshold method are equal in coverage for the moderately correlated data, and the threshold method is performing slightly better than the highly correlated data. We're hoping that we can get some of these multiple imputation methods to have that 95% coverage that we kind of look for as statisticians to say that this is a valid method to use for imputing missing data. So that's all for me. Thanks for listening.
Thank you, Suni. That was a really great presentation. Good afternoon, everyone. My name is Cynthia Fisher, and I'm a statistician in the Division of Analytics and Informatics within the Office of Biostatistics at FDA's CDER. And today I'm going to speak more directly from review experience. I'm going to focus on the patterns we see repeatedly in submissions that involve digitally derived endpoints, the gaps that generate reviewer comments, and what sponsors can do differently. Next slide, please. So before I get into any specific challenges, I sort of want to set the context for where these observations are coming from. As reviewers, we encounter DHT-based submissions across a wide variety of regulatory pathways that you've probably heard about more than once today. You have your traditional INDs, NDAs, BLAs. You have drug development tools through the DDT qualification program. You have ISTAN submissions, and the list goes on. But what's worth noting is that despite the variety of pathways, the statistical and methodological issues we see are pretty consistent across all of them. We're seeing the same fundamental gaps appear fairly regularly. So the lessons I'm sharing today are not really pathway specific. They're pretty broadly applicable regardless of how a sponsor is engaging with FDA. So this session is framed specifically from the reviewer perspective. So I'll be talking about what we expect to see when we open a submission, what's commonly missing, and how those gaps get communicated back to sponsors, which usually means deficiency letters or information requests or just more delays. So I chose the most common issues that I've seen with the hopes that this is helpful for future submissions. Next slide, please. So the first challenge is one of the most common sources of deficiencies that we see: algorithm validation in the wrong population. So our expectation is that the algorithm that derives your endpoint must be validated in a population that's representative of who will actually be enrolled in your study. So this is where disease severity matters, age matters, movement characteristics, whether people have tremors or fatigue. All of these things affect sensor signal and algorithm performance in ways that can be substantial and clinically meaningful. But what we frequently receive instead are algorithms that are validated in healthy volunteers or in populations that are demographically distinct from the intended study population. And we sometimes see key subgroups like specific age ranges or disease severity strata actually being excluded from the training data entirely. And the assumption appears to be that if the algorithm works well in one group, then it generalizes. But that assumption is not supportable without evidence. And this also matters a lot because an algorithm that performs well in healthy adults may actually systematically misclassify signals in patients with altered gait or, with severely reduced physical activity. And that misclassification can then bias the endpoint in ways that are extremely difficult to detect or even correct after the fact. So by the time we identify that there's a problem in the pivotal trial, the damage is already done. So I think it's important to understand that validation isn't this one-time event that sort of travels with the device. It's population specific, and it needs to be demonstrated in the people you're actually studying. Next slide, please. So this slide shows some examples that provide a general representation of the types of comments we might provide for some of the gaps I just described. I'm not going to read through them all, but they do express the same idea, that the algorithm needs to be validated in a data set that's representative of the intended clinical population. Next slide, please. The second challenge is missing data, specifically the failure to pre-specify a handling strategy at every level of the data hierarchy. As we've seen today, DHT data is inherently hierarchical. You have epochs that roll into days, and then days that roll into periods, and periods that roll up into subjects. So a complete strategy needs to define what constitutes a valid observation at each one of those levels. Submissions routinely arrive with missing data criteria that are vague or inconsistently applied across visits or specified only at the subject level, which leaves everything below it unaddressed. And that ambiguity is difficult to resolve after the fact, and it sometimes raises legitimate questions about whether the handling of missing data is what shaped the results. Another often overlooked concern in DHT studies is that missingness is often not at random. And Suni did a really great job of explaining this. But what we see in the real world is that patients tend to remove devices on days where symptoms are most severe. So for example, if you have a patient with nocturnal scratching, they might find the device just completely uncomfortable or unbearable on a particularly bad night. Or if you have a patient with very low physical activity, they may just not want to wear the device at all on their worst day. And what that means is that a standard complete case analysis will systematically overestimate favorable outcomes because the worst days are the ones that are missing. So that's why understanding both when and why missing data are present is essential for us as the agency to properly evaluate DHT-derived endpoints. So we strongly encourage you to submit reason for missing this data, whether that be from subject diaries or device error codes or something along those lines, because it really helps enable appropriate sensitivity analyses. Next slide, please. Here you have more general examples of possible reviewer comments. You'll notice that these concerns address both the procedural gap that I mentioned, so that's that pre-specification at the lower levels of the hierarchy, and the analytical concerns about the missing at random assumptions. Next slide, please. So the third challenge is making sure that you include patient-centered justification for endpoint selection. And this goes back to Lily's presentation. FDA's patient-focused drug development guidances directly inform how we evaluate DHT-derived endpoints. So the specific metric chosen and any threshold used to define meaningful change must be grounded in patient input. So what we'll see is that sponsors will conduct qualitative research. So they'll do patient interviews or concept elicitation studies. And then they'll use that work to establish that a concept like nocturnal scratching or sleep quality is relevant to patients, or what we call a meaningful aspect of health for patients. And that work does a good job of answering the question, what matters to patients? But there's a second question that also requires patient input or should have patient input. And it's one that sponsors sometimes overlook. And the question is, well, how should we measure that? We see that a single wearable can generate multiple possible endpoints. You could have scratch duration versus scratch frequency versus scratch intensity. And each of those captures a different dimension of the same experience. So the choice among them requires patient and caregiver input to establish which representation directly reflects patient experience and what they would want the treatment to change. So selecting a metric because it's analytically convenient or because you know that the device measures it well is often not a sufficient justification, and that's the gap that reviewers will identify. So this is where usability testing or cognitive interviews are really important because they help confirm that patients can actually engage with the device as intended in their daily lives, and that what the device captures reflects their experience. Next slide, please. Again, here are some more example comments. And as you can see, patient meaningfulness needs to be explicit, and just citing literature is often not enough. Next slide, please. Okay, so this fourth and final challenge is actually a very common misconception that we see. It's the conflation of device clearance with endpoint validity. But first, I think it's important to state that a device does not need to be 510(k) cleared to be used in a clinical trial. Clearance is a marketing determination made by CDRH. It's not a prerequisite for investigational use. We see that sponsors sometimes assume that using a cleared device sort of resolves their evidentiary burden in a drug approval context, but it unfortunately does not. Secondly, even if you are using a cleared device, clearance and endpoint validity are separate regulatory questions. Clearance establishes that a device measures something accurately under the conditions and indications of that cleared submission, nothing more. It doesn't establish that the device is fit for purpose as the basis of an efficacy endpoint in a different population under a different protocol using a different clinical construct. So both the DHT, so the device itself, and the endpoint it generates must independently be demonstrated to be fit for purpose, and that demonstration needs to be made in the context of the current program. A related issue to this is generalizability across programs. We'll sometimes see sponsors cite validation work from a prior trial as sufficient justification for the current one, but unless the populations, the conditions of use, and the endpoints are sufficiently similar, and that similarity is explicitly justified, prior validation doesn't just carry over automatically. We still have that evidentiary bar that applies every time. So next slide, please. Here is my final set of comments that are representative of what we could send to applicants. You need to establish that your device and your digitally derived endpoints are fit for purpose, and a clearance is most likely not enough information to establish that. Next slide, please. So just to sum up, whether the device can measure something is only part of the question. The other part is whether what it measures can support a regulatory conclusion. The good news is that every gap I described here is preventable. The sponsors who avoid these deficiencies aren't really doing anything extraordinary. They're validating in the right population, they're pre-specifying at every level, and they're anchoring their endpoints in patient experience. And perhaps most importantly, you've heard everyone say this a lot, they're engaging with FDA early. But we keep saying this because early engagement raises these concerns when there's still time and flexibility to address them, so before they become these deficiencies. Next slide, please. These are the references that I mentioned during my presentation. And yeah, that's the end of my talk.
Thank you both for those presentations. Now I would like to introduce our panelists who will be joining us for the discussion. First, we have Devin from Merck Research Laboratories, then Jess from the Center of Biologics Evaluation and Research, CBER, here at FDA, will give her thoughts. Devin, I'll hand it over to you for your opening remarks. Thank you.
Thank you, Laura Lee, and thank you to the organizers for giving me the opportunity to participate as a panelist. Just by way of background, at Merck, my role is to lead a group of statisticians and data scientists who support projects all the way from drug discovery through clinical proof of concept. We do have a small team that works closely with other subject matter experts on DHTs. And really, as hopefully as has come across in the entire day thus far, even though the session is about statistics, really it's the joint work with other SMEs and the statisticians that really makes everything work nicely. So within Merck, our current focus really has been on exploring whether conventional phase 3 registration endpoints can be replaced with fit-for-purpose digital endpoints. And I know this expression has come up repeatedly throughout the day, I think for good reason. But really, fit-for-purpose digital endpoints, importantly for more efficient go, no-go decision-making in mid-stage development. So I know much of the conversation thus far has been on maybe trials for registration, but my brief remarks and as of now will focus on if you are developing a novel digital endpoint, perhaps as a replacement for a more conventional endpoint that is likely to be needed in phase 3, kind of working backwards from there, how could you use this intelligently in a fit-for-purpose manner in mid-stage development, to make a proper go, no-go decision? So of course, in that context, you have to think about what are the fit-for-purpose attributes of a novel digital endpoint. I mean, the ones that come to mind immediately are obviously it has to be clinically relevant, meaning it needs to capture meaningful aspects of the patient experience and the disease trajectory. It has to be informative. And what do we mean by that? Oftentimes, it's going to be a composite measure that's going to put together different features. And so really, what we mean by informative is it has to properly integrate non-redundant digital features. I mentioned a minute ago that it has to be, if you can use this in a mid-stage trial, maybe for a more efficient go, no-go, then clearly this novel digital endpoint needs to be aligned and precise. And what we mean by that, so aligned is, it has to be directionally aligned with the conventional phase 3 endpoint, minimally at a summary statistic level, but ideally at a patient level. And obviously more precise, meaning it needs to have, notably lower variability so that you can make the case that the reason we are using a novel digital endpoint rather than the phase 3 endpoint in the mid-stage trial is perhaps we have an opportunity to make the right decision, but with a novel endpoint because it has much smaller variability, but it has all the other features that you would want to see. Clearly, it needs to be generalizable, so it needs to perform consistently across studies and across populations, and this point has come up repeatedly earlier as well. And of course, perhaps the most obvious one is it has to be predictive. I mean, it has to be reliably able to predict phase 3 success or failure with the conventional endpoint. There are obviously some statistical considerations that one needs to keep in mind. And certainly as we have worked with other SMEs at Merck in this context, some of the considerations that come to mind are things like feature selection. So again, there was use of machine learning tools. Oftentimes, you might start out with a device that is measuring maybe 100, 150 different features, but you might have a training data set that only has maybe 80, 90, 100 people. And so clearly, it makes sense to use these machine learning tools that are specifically designed for these settings. And so again, using supervised machine learning tools, we can help identify the most informative digital features. We all know in this context, there might be excitement about using and creating a novel endpoint. And so one has to be careful about overfitting. So this is where, again, if you're familiar with the machine learning space, one of the central tools in there is cross-validation. So that is an incredibly important tool to leverage to obtain realistic estimates.
Thank you, Devin. And I know I want to make sure that we have some time for Jess, and if you have other comments, maybe we can work it into some of our Q&A. Thank you.
Thanks.
Jess, over to you.
Good afternoon, and thanks, Laura. As a reviewer, I really appreciate the patient-centric justification for endpoints that Cynthia spoke of and the missing data mechanisms that Suni described. From my perspective, it's also important that the DHT data is prepared for regulatory use early in development. So in addition to that fit-for-purpose evaluation, it's important to determine how much data is needed for this reliable estimate and really have a defined acceptable threshold for things like wear time and signal quality. It's those thresholds that can support alerts that can identify problems early and really allow sites to troubleshoot or provide device replacement. What we want to avoid is discovering late in the trial that a device malfunctioned or the validation was insufficient or there's too little usable data collected. If the shared goal is really to ensure that the data collected is reliable, it's traceable, it's clinically interpretable, it really is suitable for the intended regulatory question. Back to you.
All right, thank you all for that. Thank you both, excuse me, for your opening remarks. So we will transition into the discussion portion of our session. Can the rest of our panel come on camera? And Suni, maybe I'm just not seeing you on my screen yet. Oh, there you are. Okay, great. So for those of you all in the audience, if you have questions, please put them into the Q&A. Thank you. So let's start with the question, what challenges could be avoided or caught early in trials well before the data comes in to the FDA statistical reviewer? So Cynthia, I will start with you.
Sure, that was a great question. A few challenges stand out to me. First, device compliance in this at-home setting. We sometimes see patients forget to charge their devices or, like I mentioned, remove them during their worst symptom days or just wear them incorrectly. And unlike during a typical clinic visit, there's no staff there to catch that. As I mentioned in my earlier talk, that kind of missingness is often not random, and it can systematically bias your endpoint in ways that are very hard to recover from analytically. The other challenge that comes to mind is operational inconsistency across site. So things like standardized training on device placement, charging schedules, troubleshooting, all of these aspects are very important, but often underdocumented. So we see that when sites handle things differently, you introduce variability that may look like signal, so it might look like treatment effect, but it's really just noise. But these are all prospective problems with prospective solutions. So if you can build those solutions into your protocol before you enroll any patients, you can avoid a lot of these problems down the road.
Do others have thoughts? I don't know, Devin?
Yeah, I guess I could offer maybe 3 points. One is, to define the digital endpoint and the estimand clearly before the study start. And, the more clarity there is in terms of how that raw device-based data was transformed into the analysis endpoint, because that's a critical journey that needs to be fully transparent. How that is going to be done and the clarity around that is critical. And the other two are maybe sort of obvious, that to monitor data completeness and adherence in near real time, rather than waiting until database lock. And again, hopefully nobody waits till the end. And third, we do this even for non-digital endpoints where we anticipate there's going to be missing data. Why the data are missing and the reasons for missing are really important to document. So for the non-digital endpoints, we often work really hard to understand if somebody has dropped out. Did they drop out because of toxicity? Did they drop out because lack of efficacy? What was the reason for dropout? Likewise, in the data collection process, and well before the data are analyzed, in the database, as best as we can to document why the data are missing, I think is critically important.
Excellent points. Suni, do you have anything to add?
I would probably say that not only should we be documenting the missing data and the reasons for it, but having some strategies for how we would kind of augment that missingness, what types of imputation we'll be using, and how that would affect our results or estimates that we derived.
Right, as long as I'm going around, Jess, yes.
I see another challenge to DHT-derived measurements is simply life happens outside this controlled clinic environment. So the real world environment can be noisy, can be unpredictable. You can have a heart rate increase that might reflect the illness of a treatment, or may completely be unrelated to the condition of the treatment. And we recognize that environmental conditions and everyday activities can affect DHT measurements. So I think sponsors should consider collecting relevant contextual metadata and pre-specifying how that information will inform the analysis rather than trying to reconstruct that context afterwards. And I think what a lot of people have already alluded to is protocol deviations related to wearables are much less visible outside the clinic. So a wearable may be positioned incorrectly. It could be used inconsistently, can malfunction without the site team realizing it. So I think sponsors should consider deploying like a risk-based remote monitoring with having some automated data flow alerts and structured patient participant check-ins so that the data quality issues surface early enough to be addressed, not after the database lock.
Oh, thank you. And I think all of this really important issues, and as was brought up, many of these are things that come up outside of our digitally derived endpoint area, but it becomes even more important as we're using technology and many times where things are new to a lot of new people. So I'm going to turn us a little bit to that small study rare disease space. Do you all have any recommendations for designing or analyzing small studies, for example, in the rare disease setting, so that the within patient digital health technology evidence, that information that's coming in on the DHT, is leveraged without overstating a treatment effect? Do folks have thoughts on that? Devin, maybe you do?
We don't do rare disease. At least our experience in this context is not with rare diseases. But certainly, as I mentioned earlier, we are focusing on, can we, instead of say needing 200 or 300 or 400 people for a mid-stage clinical trial with a conventional endpoint, could you maybe cut that sample size in half, meaningfully? Now you're entering a relatively small data set situation. And so now, all the usual aspects that come into play. At this point, are you worried about p-values only, or should you really think about, do you need to focus also on estimation? Is there an opportunity maybe to use some Bayesian principles so that, again, not being wedded to p-values and confidence intervals with small sample sizes, could you actually do something ultimately to say, okay, well, based on what we're seeing with this, in this case, this novel endpoint in a relatively small sample size by construction, because that is what you had claimed, that you could make a decision with a smaller sample size. But again, are you confident that this will translate into success in phase 3? And the only other point I'll make, when the sample sizes are even smaller than the ones that I mentioned, perhaps in a rare disease setting, there's a temptation to say, well, I only have 20 people in my trial, so instead of measuring, 500 observations, I'll measure 5,000. And I think people need to appreciate that there comes a point where just simply measuring more and more data on the same patient is not the same as having more patients.
A really important point. Do others want to add on to this or provide your own perspectives? No, Cynthia or Jess? Go ahead, Cynthia.
Yeah, I can speak a little bit to this. I've worked a little bit with SV95C and Duchenne's muscular dystrophy, so that's sort of my rare disease knowledge. But just to echo what Devin said there, I think sometimes we do, just with the sheer volume of data that DHTs provide, we sort of sometimes see this false sense of statistical richness when your trial only has a small number of patients. Because you have the ability to describe what's happening in those patients very precisely, you have this sense, but the ability to generalize that to a broader patient population is still limited by how few patients there actually are. Both those things matter, but it's just important to be honest in your analysis and interpretation about what kind of evidence you're actually providing. As far as recommendations, I would say pre-specify your meaningful change threshold before you look at the data. We've seen that sometimes in small studies, there's this urge, whether it's conscious or not, to let the observed effect size inform what gets called meaningful, but that's a form of overfitting, and reviewers will notice. Particularly in DMD, we see leveraging of natural history data where it exists. We often have these well-characterized trajectories of decline from registries or observational studies, and that external benchmark is very valuable because it gives you a comparator when a concurrent control arm might not be feasible. And it helps you distinguish if the true treatment response would have happened anyway. Compare that true treatment response from what would have happened. But you still need to use that very carefully and transparently. And like we keep mentioning as well, pre-specify your missing data rules. When you have a really small N, even 1 or 2 patients with substantial missing data can significantly shift your results in a way that would normally average out in a larger trial. So those are my 3 recommendations.
Those are great recommendations. And I think an important part is, like many times groups are trying to figure out how to have that smaller sample size, even where it could have been and normally would have been much larger. But this idea that we really do need that independence, that, one person or a handful of people cannot be everybody. And that, especially as regulators, and really for science in general, we want to understand that we have reasonable generalizability and a nice variety of data and information to pull from. So with that, I'm going to move to another question. I know some of this is missing data, then we'll move a little bit past missing data. But when does missing data really become a problem? And I think I'm combining a few questions also that we have from the public here. So when is it that there is still enough information to trust the results? And when is it, and it might be kind of within a person, across people, within a session, not within, there are different ways that people have phrased this, but when in your experience has this become a problem? And then when is there really enough data and information we can trust those results? I know, Cynthia, I know you just spoke. Maybe I'll go ahead and turn back to you. And Suni, you brought up some really interesting examples, and then we'll open it up to others.
Sure, yeah, no problem. This is actually an issue that I've discussed with my colleagues a number of times. And what we always keep coming back to is how important it is for FDA to understand why the data is missing. So a small amount of missing data that is truly random. So you have a device malfunction or a transmission error that's unrelated to how the patient is doing. That kind of stuff is generally manageable. But as I've said before, missingness is rarely random. We see the patients remove the devices on their worst days. So when that happens, even a moderate amount of missing data can seriously bias your results, because the observations you're missing are the ones that would have pulled your estimate in the unfavorable direction. So the question I'd encourage sponsors to ask is not how much data is missing, but who is missing it, and when are they missing it, and why are they missing it? And that means actively collecting that information. So that could be device error codes, subject diaries, site logs, and build that kind of stuff into your protocol from the start, because that is what will help tell you, if there's enough information to trust your result. And what makes us more comfortable, even with moderate amounts of missing data, is pre-specified sensitivity analyses that sort of stress test the results under different assumptions about why the data are missing. And if your conclusions hold up under those scenarios, that's pretty reassuring. But if they don't, that's also important information, because it's better to know that than to present a result with false confidence.
I think I'll probably just add that it's probably important for, at least with very significant disease populations, to actually do a pilot study in those populations and trying to figure out maybe getting rich sources of data in the pilot study to understand more about what causes missingness in the data set and how much missingness is acceptable and how much of it biases a result that you're interested in showing or interested in testing. And so for us, doing sensitivity analyses are always important, doing pilot studies and even simulation studies with the pilot data to see how much of a kind of I want to say how much missingness will allow you to still recover the same kind of estimates that you would see in complete data.
Do others have thoughts?
Sure. Yeah, this is Devin. So the only other thing I'll add is there's much to be learned from non-digital endpoints in this missingness discussion as well. And specifically, what I have in mind is in clinical trials with non-digital endpoints, quite often we actually find out that the people who are dropping out tend to or have missing data. They generally tend to have something in common. And so what is that common denominator? And so it might be, for example, it might be people who are older or who have higher disease severity at baseline. They are the ones who are more likely to have missing data. And so if there's any way sort of at the study design stage to anticipate that perhaps there's certain, it's a guessing game at that point, but hopefully an educated guess. If you can zoom in on who are the types of patients in your trial who are more likely to have missingness, perhaps you could leverage that information up front in the design and conduct of the trial.
Any other thoughts? All right. Can you provide an example where a sponsor or you yourself prepared for a software or some other type of update during a trial or moving between trials? I think something that's been brought up a few times and that folks have definitely had some issues with is like technology is always changing, right? So how do you pre-plan for those types of things? And maybe you can't talk about that, but kind of what are some examples where you've had to handle an update during a trial or as you were moving from a phase 2 to a phase 3, something like that? Devin and Suni, I'm going to maybe go to you all for this, but if others want to chime in, that's fine too.
Sure, yeah. So thanks. The example that comes to mind is a few years ago, we were developing a drug for chronic cough. And to measure chronic cough, there was a device that had to be used. People had to wear the device that was recording. And of course, it was recording not only the coughs, but there was other noise, background noise, and so on. And so you can imagine you have a 24-hour recording from a wearable device, but in the end, how do you determine, at baseline and then post-baseline, how many times is the patient coughing? And so there was an algorithm that had been developed to basically take this 24-hour recording, and then the algorithm would then remove the noise and hopefully retain only the coughing episodes. And the coughing episodes then could be counted by human counters. So this was a very interesting situation where the algorithm, in this case, it evolved over time. And so that really raised the question as to, do the differences in the endpoint values reflect treatment effects or changes in the endpoint derivation as a result of the evolving algorithm? So there were some lessons learned here, and certainly, this experience reinforced the importance, obviously, of retaining the raw data, in this case, the entire 24-hour recording, maintaining version control, in this case of the algorithm, and performing comparability assessments whenever algorithms are updated. So of course, this was an example with an algorithm, but the same would apply if you have a device that's being run on, maybe an iOS platform or some other software. And as that gets updated, one needs to understand upfront as to what is the impact going to be on the endpoint.
I think for us, when the QSU was working with the Apple Heart Study, we came across an issue that is called deduplication. And it really had to do with the fact that people were registering their devices multiple times, registering new devices. There were software updates happening. And so the data streams coming in looked like they were from a lot of participants and a lot of devices, but we had to find a way to kind of create a unique set of participants and devices that belong to each participant. And we used a method called entity resolution to do this. And so one of those questions is kind of making sure that you have that data flow mapped out correctly, and understanding where like data is coming into your study from and how it could create issues with identifying participants.
Great. Thank you all for your thoughts on that. I did want to ask a new question, and I think, Devin, in a way, you've already maybe addressed this a bit, but a lot of people are asking, is there a way how to help explain that missing data? And so some folks have asked, like, can you kind of triangulate or use that information from patient diaries or ePros to help explain the missing data? And do you all have any recommendations of things that sponsors should consider as kind of their companion methods, let's say, for data collection along with the DHT? So how are you seeing that folks are actually getting to this information? Do folks have any recommendations or thoughts on that?
Sometimes I think the sensor can actually help you. There might be kind of auxiliary information that the sensor has that will help you determine why something is missing. With the screnomics data that we were looking at, we could tell when the battery on the device was charged or not. And so that would give us a sense of like, oh, is the missingness caused because, this person hadn't charged their device overnight and had lost battery on it. Things like that might be able to help. I get slightly concerned with patient logs in terms of increasing patient burden. And I don't know if by increasing patient burden that would lead to more missingness in your data rather than kind of helping you resolve missingness data issues. So finding a balance between the two.
Any other thoughts on ways to help collect the reasons for missing data? And I do know we spend quite a bit of time trying to triangulate information from, it might be exit interviews or pre-interviews from things that we even learned during the development period, whether it's for the DHT or anything else. And so it can, and sometimes it is picking up the information to look. But I take your point that sometimes we're asking to collect extra information, but it can be difficult. But to not forget all this extra information from the sensor. Oh, Jessica, maybe you have some additional thoughts, or Devin.
I think the beauty of DHTs is you could set algorithms for thresholds, right? You can monitor, and I know sponsors hate when I say real time, but you can monitor, pretty much in real time if you have the resources. And catching that early is so invaluable. Right? Catching, well, this person failed to have this reading or did not enter their e-diary, catching it early, getting to the patient, what happened, getting that contextual data around it is really helpful. Having the, ECOA, like you said, will help us understand irregular data, but missing data, you just have to get earlier rather than later. And by the time you have a clinic visit, that might be too late.
Yeah, I do know it's so important here that kind of this isn't kind of audit trail information at the way end. You want to be paying attention to a lot of information early and making adjustments, kind of doing that piloting early on. And that usability data, as I mentioned, can also, doing the usability studies is really important to then, it's not separate in some ways from your clinical trials. It really does many times help us interpret what happens in the clinical trials. I don't know if others have thoughts. Have you all ever seen kind of maybe like small pilot studies trying to figure out ways to maybe assess or process or reduce missing data? I do know in some clinical areas we maybe have seen some of that, but I don't know if that falls where you all are. But I would say this is one of the great things that happens with our DDT programs is that there is a lot of collaborative learning and learning in phase 2, not just about your products, the medical products, but also about your assessment methods. And then that helps you in your planning phases. So I want to quickly go to 2 last questions here. And one, putting aside the missing data part, although this may tie to what you want to close with, I don't know. But putting aside missing data, what are the few of the other challenges that you think stakeholders should consider? Jess, you were just talking. Do you have anything else? Cynthia or Suni? Go ahead.
Yeah, I can jump in real quick. One thing that came to mind with this question is behavioral influence. So we see that when patients know that they're being monitored by a wearable, especially one, to your point, Jess, sorry about this, that gives real-time feedback, it can change their behavior. So a patient who can see their step counts might walk more, or a cough patient who's aware of the throat sensor that's on them, they might try to suppress their coughing. And that's not really a data quality problem in the traditional sense, but it can still distort your endpoint. So this is where blinding decisions and whether feedback features are enabled need to be carefully thought through upfront. Another thing that we see could be environmental or seasonal confounding. I know that in physical activity and sleep and even scratching behavior, I think it can vary meaningfully by season or climate or day of the week. So when you have a multinational trial that's running across different geographies or time zones, those factors can introduce variability that has nothing to do with the treatment. And sometimes we see that that's not addressed in the analytical or analysis plans. So that's something that is important to note.
Suni, did you want to add something?
No, I was just thinking that in some populations, especially low resource populations, things like Wi-Fi connectivity and making sure that your study with the digital health technology isn't adding kind of an expense to the population that you're looking at. I had a friend of mine who was doing a study in indoor air pollution, and he had sensors kind of wired into the participant's home, but he also took the time to help pay their Wi-Fi bill so that wasn't exceeding kind of data limits that they had. So learning about your population and what they can do, what you can reasonably expect from them.
And that is a really important point. And I will say that those accessibility issues, but also natural disaster issues that you sometimes have to plan for, but really making sure that the participants you want in your clinical trials can participate in your clinical trials. And that's an incredibly important part of making sure that we get the data that we need and that the trials are going to be successful. So we're getting close to time. I'm going to say 30 seconds or less. I'll give you a few extra seconds if you need it. What is your number one best practice that you want people to take away from today? It may be this session, it may be another session, but what's the number one best practice you have that you want people to use to help ensure consistent, rigorous evaluation and to address the statistical and analysis challenges that you all are witnessing. So I'm going to move us quickly through here. Suni, Devin, Jess, and then we'll close with Cynthia. So Suni, number one best practice.
I always say the number one best practice is a collaborative interdisciplinary team. Our research takes data engineers, subject matter experts, statisticians. There's a whole group, even patient advocates can be helpful. And having a team that's willing to talk to each other, troubleshoot problems with each other, kind of makes for better research.
Great. Devin.
Sony took mine, so I will say ditto, but also the cross-functional alignment, not only between digital measures, operations, stats, within the sponsor, but also between the sponsors and the DHT vendors is very important. And then also, again, what I said in my opening remarks, we really need to very carefully validate the entire measurement pathway, from device-based data collection to the analysis endpoint derivation. That fully understanding that journey is just critical.
Absolutely. Jess.
So I'm going to be at risk for sounding like a broken record, but I say pre-specify everything before enrolling your first patient, locking your endpoints, your algorithms, your missing data plan in advance. This can create a transparent record against which the final analysis can be evaluated. It also helps determine whether the findings are robust and it's driven by missingness or these analytical choices. So, of course, like if you're uncertain, engage with FDA, engage early.
All right, Cynthia.
I'm also going to sound like a broken record, but engage with FDA early. Engage with us earlier than you think you need to. Like I said in my talk, the sponsors who avoid the issues that we've all been mentioning are the ones who are coming to the table early, coming to the table while there's still time to course correct. We see that, when a full submission arrives that has a fundamental gap in algorithm validation or endpoint justification, the options for fixing it are very limited and expensive. So when you have early engagement, we can help raise those concerns when there's still time and flexibility.
Yes, absolutely. And I will say sometimes what you're hearing today, what you read in guidances, what you hear from FDA and other regulators, it may seem like a lot, but what we're really trying to do is avoid problems that we have seen and get to positive solutions and get great medical products on the market. Thank you to our presenters and panelists for a great discussion. This is all the time we have for today's session. We have one more session to go and some closing. So now we're going to have a short break, though, before returning to our last session of the day. Thank you again, everyone.
Hello, everyone, and welcome back. I'm excited to be moderating our last session of the day, where we will be exploring emerging opportunities, challenges, and priorities, shaping the future development and regulatory acceptance of digitally derived endpoints. Today's discussions have highlighted FDA's commitment to collaborating openly with patients, scientists, and industry leaders to advance innovation while upholding rigorous evidentiary standards that protect patients. Looking ahead, we want to explore key challenges, shared priorities, and emerging technologies, focusing on how these developments will shape the design, evolution, and regulatory acceptance of digitally derived endpoints. I'm now going to ask all of our panelists to come on screen for the session. First, we have Gül Erdemli, Global Program Regulatory Director at Novartis. Our second panelist is Hussein Ezeldin, Senior Public Health Advisor at the Office of Medical Policy in the Center for Drug Evaluation and Research at FDA. Then Christine Gual, Clinical and Data Science Team Lead at Amitris. Gabriel Krotov, a statistician, I can say that word, statistician at the Office of Biometrics at the Center for Drug Evaluation and Research at FDA. Valentina Mantua, the Associate Director for Regulatory Science in the Office of New Drugs in the Center for Drug Evaluation and Research at FDA. And finally, but not least, we have Benjamin van der Schie. I think hoping I'm getting that quite correct. The President and Chief Scientific Officer at the Digital Medicine Society, or DIME. So, Gül, we're going to pass it to you for opening remarks.
Thank you very much. First of all, thank you for the organizers for opportunity to participate in this very interesting workshop. And so today we heard from other speakers multiple times the potential of these digitally derived measures to transform and impact the drug development by providing more comprehensive, holistic understanding of the disease and disease impact to the patients, and providing a better assessment of the new treatments in terms of their efficacy and safety, and ultimately providing patients early access to treatments where the intervention really changes the course of the disease. Having said that, on the other hand, we also heard, and we all know, these measures come with challenges. For example, extended period recordings, continuous recording during extended periods is very attractive. But on the other hand, depending on the technology, nature of the technology, and how the patient interacts with that, these could be quite burdensome, leading to poor adherence and missing data. Today we hear a lot. And also towards the end of last session, we also heard another challenge briefly, that these measures can be influenced by external factors that are not related to the patient, to their disease, not even treatment. Therefore, it's really important that from the beginning, when we are working on new measures, to identify the intended purpose, value over and above what are the existing measures and what are the challenges, and agreeing on how we address these challenges. If I have a couple of minutes, maybe just to also add that one of the important barriers from an industry point of view is a lack of global alignment when it comes to expectations for validation, interpretation, and regulatory acceptance of these measures. From FDA, we have digital health technology for remote data acquisition guidance, and also several times today mentioned patient-focused drug development guidance series. The recent white paper, these are certainly significant step forward for providing clarity in the United States, but also patients still carry the burden of getting global alignment from other health authorities, and having an harmonization could certainly push the field forward to adapt for adoption of these technologies in a broader manner, and maybe consider an ICH guideline or series of ICH guidelines to provide harmonization. Thank you very much. Looking forward to the discussion.
I am too. Hussein, can we go to you next?
Thank you so much. Very honored to be here and join this amazing panel that has a lot of expertise. It's not really a coincidence here that we have this breadth of perspective. Digital health technology developers, we have statisticians, we have science measurement experts, and I think this is the right mix that you need in the room to have this discussion. So really, thank you so much for bringing us together today. I think just continuing on what Gul mentioned, alignment is definitely very critical, as Gul mentioned, on the global space, but also even within the agency. I think we owe it to our patients, we owe it to our sponsors to have a consistent messaging across all review divisions. So I think this is something that's really important, and we are working hard on doing that. Maybe I'll speak again. Feels like I'm repeating what Gul mentioned, but I think the timing is really important. So you wanna have the right people at the right time together. And Gul alluded to the digitally derived the consideration for digitally derived measures paper that was published last week. And in it, we tried to at least do a very small effort here to bridge this gap, connect the science measurement experts with the digital health technology developers so they can start this discussion early. So I think we always say, come to the agency early and engage early. I think that the other piece that we want to also stress here is that we want the developers, we want the science measurement, we want the sponsors to also connect early and be part of this journey together to be able to produce the needed evidence for moving the field forward. And my colleague Beth Kankowski alluded at the top of the workshop today about CDER commitment for putting out a process engagement guidance to kind of help out how people can engage with the offices and facilitate these interactions. And maybe the last thing I will mention is that we are trying to leverage all the tools that we have. And Matilda early mentioned that we are trying to expand the PDUFA commitments and kind of reach out and work with DHT developers to get real data that helps us understand better these measurements. So all these are efforts that we are trying to put together here to push the field forward. Looking forward to the discussion. Thank you.
Thank you so much. Christine.
Yeah, thank you. So I'm here representing an organization that has been working on this very topic for more than 2 decades. So it is amazing to see the range of discussion and the depths that we are able to go through in the FDA hosted session workshop today. So I'm also very proud to see that many talks actually cited the use of our technology in their research. So I'm quite honored to be part of this panel to reflect on this journey that we've been and provide a bit of outlook on where we're going. I think we should be proud of the progress we've made so far, especially in the last 5 years. We talk about the alignment on regulatory agencies and the white paper that just came out that I think really provide more clarity. And as a technology provider, I think we had also really matured in the way we build the technology and the platform. Certainly that's the easy part. We've generated more evidence. And also that I think we're really focusing on trying to work out the operational kinks so that we can minimizing the missing data as we are talking about today, right, before it actually happened. And put in sort of more practical terms, I think as a technology provider, we are seeing more successes are coming out of the pipeline. Validating and developing a novel endpoint is very challenging as we talk about today. And often it's because we actually needed to be part of a successful trial with a working drug to be convinced that the endpoint is working, right? And as we know that the odds there, typically it's kind of against us. But because we've been really chipping away at this for a very long time, we are seeing those wins. Some of those had made into public domain, like the physical activity endpoint in Pfizer's cachexia program. And there are many that I think will be coming to public eyes very soon. But it has been, I think, challenging and take a while as we talk about. I do think that we need to continue to work on the mindset shift as we try to capture this momentum. Even as we, let's say, get much better at verification and analytical validation, I believe that many people still really want to see that key part of clinical validation, which require us to get a DHT into longitudinal trials and especially clinical trials. And this is where I see a lot of times the conversation kind of gets stuck with this chicken and egg dilemma, where the clinical team want to see more validation before they want to put into a trial, but we needed this data to be coming out of trial in order to get the evidence we wanted to see, right? So this is sort of where I think we need to continue to see how we can move this forward. And at Ametris, we're trying to bridge these gaps by building kind of pre-competitive multi-stakeholder partnerships. We've launched really pre-competitive evidence generation working groups, which we call Decode. We've basically been hosting our annual conferences, focusing on the science and the methodology, focusing on DHT. And several FDA colleagues came to us this year and joined the conversation. So I think this kind of multi-stakeholder, as Hossein just mentioned, is very critical on how we can move the field forward. And I look forward to the conversation today, talking about how we can further get the rubber meets the road.
Thanks, Christine. Gabrielle.
Alrighty, yeah, thank you, Christina, and good afternoon, everyone. I appreciate you all sticking around for the last session. So the topic of this panel is like the future of digitally derived endpoints, and kind of drawing on some of the lessons that Hussein referenced earlier. I think it might be valuable to highlight some aspects that can build confidence in a digitally derived measure, like especially in the analytical validity space that Christine was just talking about. The one that stands out to me first is having a really clear flow of data between the sensor level and the analysis level. Because in the process of converting between sensor level data and analysis level data, you are inevitably making a lot of decisions that are important to the analysis. And kind of like that accumulating error that Ryan talked about in his excellent earlier talk, those decisions are unavoidable. And you would rather be explicit about them than leave them implicit. And then kind of the second thing that I see being a part of successful digitally derived measures in the future is transparency about data quality and imputation. We heard a lot previously about missing data and imputation, and typically you need to meet a pretty high standard of scientific justification to modify, discard, or impute data. And transparency about exactly how you're doing it helps you do that. And so I've kind of highlighted these two aspects of successful digitally derived measures, and there's kind of this rider that comes with them. It's true that in submissions that can support those, you will have just a lot more data than people are typically used to, because if you want to support that clear flow of data between the sensor level and the analysis level and have transparency about those, you need to go down to the epoch level, which brings a big increase in data scope. And that makes cloud computing increasingly important, and cloud computing environments may have different data format needs. So it's possible that like highly efficient data formats could help facilitate review in that space and maybe break some of that chicken and egg problem, or help break some of that chicken and the egg problem. So I'm excited to get into it and hear from everybody. This is a fantastic panel.
Thanks, Gabriel. And Valentina.
Yeah. Glad to be here.
Valentina, you're coming in a little bit faint. Can you maybe lean into your speaker a little bit?
Yeah, sure. I have.
Oh, perfect.
Let me know if this works, right? So I just wanted to say that, as you mentioned before when you introduced me, I work in the Office of Neuroscience. And what came up from the talks this morning is that we've been tracking DHT submissions of across CDER, and the office that has the highest concentration of those submissions is actually the Office of Neuroscience. And the second one is immunology and inflammation, but we have the highest. And that speaks, for the need of measures in neuroscience drug development. And there's various reasons that we can talk about. It's we measure very difficult concepts, for sure, for which current outcomes aren't optimal. Sometimes trajectory is important and potentially more important than single time point. Sometimes symptoms are in isolation, and the way that they're currently measured. Instead, if you see the statistics that we have, about more than a half of the DHT submissions that we've been having are about PROs. Okay, so there's an unmet need for developing measures that matter and that complement potentially existing measures. So there certainly is an unmet need in neuroscience, and there's a lot of development there. This is why I've been interested in this field, in addition to being a technology lover, so personally. But I want to see that my role, I'm a clinician, is to look at clinical trials. And I think, reflecting on to how the clinical trial environment is changing is also important, in that we're seeing increasingly, automations of workflows, which takes sometimes, investigations away from the sites and more decentralized. So if we are away from clinical visits, then we have more time points and there's more room for DHTs and for those trajectories to be measured, I think. So that's one thing I'm seeing changing. I'm also seeing very interesting, new designs, platform studies. Sometimes, when the biology of the disease becomes known, one wants to put together, different diagnoses with similar biology. And sometimes these trials are not powered for efficacy. There is a very good place for digital health measures to be, tested and used for preliminary efficacy for de novo decisions, coupled with pharmacodynamic biomarkers. So we're seeing a lot of this. Another thing that we're working very hard in the Office of Neuroscience to foster, development of and use of technology within drug development is certainly cross-center collaboration. So a lot of these efforts that you've been hearing today, including the guidances, have been the result of cross-center collaboration. It's very timing, the key consideration for development and use of digitally derived measures for clinical investigation just came out a few days ago, and that is an example of cross-center collaboration. I think it's in the CDRH website, but trust me, there's been a lot of effort with fantastic colleagues from CEDAR on that. And I think that white paper contains pretty much all you need to know in terms of validation framework. I think it's a big step forward. It pulls away from something that we've been hearing a lot about, that a digitally derived measure needs to resemble an existing measure, which is not necessarily true. I think that paper clarifies this. There are some unmet needs for sure. One has been mentioned many times, that is, there is no optimal engagement pathway for developers, technology developers that are using their technology outside the development program, and at the same time do not want to enter qualification. So I think this is something that we're actively working on. But again, we're working on cross-center collaboration for what we can do, but really the mantra for us is breaking silos. It has to be for industry as well. Collaborations across stakeholders and potentially with other agencies. I don't have a problem, reaching out to our colleagues from other agencies and start. We have, existing, collaborations and potentially get alignment on that side as well.
Thanks, Valentina. And then Ben.
Thank you, Christina. Hi, everyone. My name is Ben Van Andries. I apologize about the last name. I didn't choose it. I'm the president and CSO at the Digital Medicine Society, or DIME, and I'm also the incoming CEO, as Jen Goldsack, who I'm sure most of you here know, will continue our mission on DIME's board as of next week. Others have already referenced the new FDA white paper on digitally derived measures. I think I do want to touch on it again, because the most important takeaway for me from that paper is that it's underwritten by the 3 centers, right? And I think that's a really positive signal about alignment that is starting to form, and others in their opening remarks for this session have already touched on that. And I think further good news is that what the paper describes is heavily grounded in approaches that have already been championed, not only by the agency itself, but several other organizations, including us at DIME. And specifically over the last couple of years now, a highly methodological approach has emerged on how to implement digital endpoints in clinical trials, where we are starting to define a process that feels less like art and more like science. I'm obviously not talking about like the science of validating everything that we do, which has already been touched on by many speakers before, but the higher level process of actually figuring out who needs to come around the table to make sure that an endpoint makes it into a trial. And what that process high level looks like, which again is highly similar to what's described in the paper, is that you have to start with meaningful aspect of health, right? We all know something a patient wants to improve, prevent, or not make worse. We need to be able to associate meaningful aspect of health with a concept of interest, meaning something that derivable from the meaningful aspect of health that is measurable, and that turns into an outcome that can be measured with some specific digital health technology. And that ultimately can become the endpoint. I think we're all aligned on that, but the DHTs that are then used to measure such an outcome, they need to go through proper verification and validation, specifically verification, analytical validation, clinical validation, and sometimes forgotten usability validation. And again, I was very happy to see that the paper addressed that. That's an approach that many of you are probably familiar with the V3+ framework, which is an approach that DIME has championed for a while now, that provides a coherent method and framework to make all these different components fit together. And analytical validation often still remains the most tricky topic because it is so highly dependent on having a good reference measure available or ground truth, right? And if your reference measure and whatever you're trying to validate captures the same construct, it's typically fairly straightforward, right? From a statistical perspective, we figured that out. But if the measure is novel and no good reference measure exists, again, as we've heard today, it gets trickier. One of the other resources that we've developed at DIME is essentially a flowchart that steps you through various levels of reference measure rigor to allow you to make an informed decision about which reference measures are available and which one to pick under what circumstances. And it also addresses the case where there are simply none available, right? And not that there's a magic solution available, unfortunately, for that yet. We're going to all collectively have to continue to work on that, but there is a framework already in place that allows us to communicate about this, which is important. The reason why that is becoming increasingly important, well, it's always been important, but right now it's becoming increasingly important, is because we have that alignment happening across FDA centers, across sponsors, across DHT developers. That means we need shared methodology, right? We need shared terminology. And the more we're going to be able to settle on that, the more we're going to be able to stare and compare across studies, across trials, and learn what works and doesn't, right? And we at DIME recently went through a huge effort to pull all the existing resources that cover aspects of getting a digital endpoint into a trial, pull all of that together into a roadmap style resource. We call it the SDHT, so Sensor-Based Digital Health Technology Adoption Navigator, or that's a long phrase, NAVI for short. And the roadmap also highlights where points of friction and uncertainty remain and where regulatory engagement may be necessary to generate clarity. And that's including one that's around analytical validation. And it's that continued engagement of DHT developers, of clinical trial sponsors, and FDA, of course, around these types of resources that is making it increasingly easy to identify where points of friction remain, what hurdles we still need to tackle as a field to accelerate the responsible adoption of fit-for-purpose digital measures and endpoints in trials. So from that perspective, I think, again, the fact that that methodological approach is emerging and is clearly being adopted by many of the stakeholders that we need around the table to make this possible is a very positive sign.
Thanks, Ben. I'm so appreciative of all of y'all joining for this panel, because I think you are all exactly the right people that I want to have answering these questions. And I think I really want to start with the vision question. What do we actually imagine clinical trials looking like in a space where digitally derived endpoints are being used commonly? How is that going to look different from today's trials, today's types of trials? And really, what is the single unresolved obstacle between that vision and where we are right now? And that's a big question. So I am going to start with Hussein and put you on the spot.
Oh, thank you so much. And this is great. I wish I was very excited if you'd called on me, and I'm grateful for this. This is the future, so I perhaps have the chance to dream and speak what I think might be the future in few years. What I envision in 5 to 10 years would be a clinical trial ecosystem that digitally derived endpoints or measures leveraging digital health technology is not really an afterthought. It's something that is included from the design stage. You're able to bring together all your DHT developers, your principal investigators, your sponsors, all of them from day one, and even the agency, the regulator as well, in the design stage, to be able to all agree on what are the data that you need to collect to actually achieve the endpoint that you're interested in. And maybe just to touch upon something Christine mentioned in her opening remarks was about the entanglement or the dependency of an endpoint or the use of a DHT on a success of a trial. I think that the future should not be this way. The DHTs, like any other technology, is a tool, and the tool doesn't have to be dependent on the success of the trial. The tool could be a great tool. It is actually capturing what we are expecting, but the drug itself might not be succeeding, and we shouldn't really entangle those together. So I hope that in the future, DHTs are not thrown at the bottom of the exploratory list, and which also leads to the collection of data that we were not part of the discussion. Perhaps even the developers themselves were not part of the discussion with the sponsors, with the CROs about how they would like to use this. And then all of a sudden you have an evidence package that you're bringing to the agency, and you get disappointed that the data that you collected is not the data that you need to hit all the marks related to proving a meaningful aspect of health or analytical validity or clinical validity or even usability. So I think this is really important. And I was looking at perhaps I was trying to make my point. I found a study by the Tufts Clinical Center for the Study of Drug Development, and they worked with transfer rate to find that close to 30% of the data collected in clinical trials, phase 2 and phase 3, are non-core data. Imagine that you are collecting 1/3 of the data that you don't really intend to use for secondary or primary endpoints. And if you're able to strategically collect this data in a thoughtful way, I think you're going to have a lot of efficiencies. You're going to be able to reduce the burden on the patients, on the trial staff themselves, and also get the data you need for a fast and efficient review and approval. Yep. Turn it back to Christine.
And since you mentioned Christine's answer, I'm going to throw to her next.
Yeah, thanks, Christina. So yeah, I totally agree that I think especially for us who've been working on this so long, it would be very exciting to see the DHT really move into the mainstream of data collection and tools for helping us. And it should be, right? Like, I think we really can see that the data coming from DHT being objective, being continuous, and it held great potential. I think that one of the things I would love to sort of see is also kind of really thinking about design trials with the continuous data collection in mind. In some way today, I see that we are writing DHT into a schedule of assessment, a protocol that's really designed for discrete, data collection, as well as the SAP, right? Like, we're trying to fit this continuous data into a framework that we're familiar with episodic data analysis. I think that for us to, in the future, especially as Valentina mentioned, in neurodegenerative diseases, to really leverage the power of some of this continuous data, perhaps we can really think about statistical methods that is suitable or designed to address, to work with this continuous data. And then the other point, other question you asked, like, what is that one thing that, in between us to this exciting future? I think there's many things, but for us to see a very meaningful shift and sort of see this future, I do believe that we have to act today. One thing I see a lot is that study team, being, let's say, working in the same way to design clinical trial, tend to be very conservative and reluctant when it comes to new things, right? There's tend to be a lot of fear about, is this going to somehow, make it challenging, compromising my study operations and etc. I think that even though today we are still talking about challenges, I feel, as I mentioned in my opening remark, we have made a lot of progress. I think we have figured out the right way to collect data, the right way to build the evidential, like a validation framework, as FDA have pointed out what is the right way to do it. So I hope that many of the study team today, perhaps thinking about it, but still feel like, okay, I've never done this before, get a bit more confident and be assured that, especially working with a DHT provider that has done this before, we can really be the partner to kind of help you navigating the road so that we can really get to that exciting future in 3 to 5 years.
Yeah. So it's kind of a combination of needing to be able to de-risk it, but also open space for that, a little bit more risk taking as well. Valentina, can I pass this question to you? What is your vision for what things will look like in 5 to 10 years if things go well, and what's the big thing we need to make sure we get right to make that happen?
Well, I said a little bit of this already. I am seeing clinical trials changing and becoming more decentralized, more community-based, certainly getting more into a pragmatic form, getting some of their workflows automated. What we have seen happening in the last few years, I mean, just think back 10 years ago, patients are increasingly more empowered. And if you couple these two trends, measurement will eventually come from more the patient in his natural environment and in a continuous manner. So that certainly is a trend that we can already see. We're trying to make it regulatorily acceptable. We're working a lot on innovative designs, but for sure there's that trend. How would that work? I mean, I would just pick on Christine's thought about we could, for us, it's very interesting to understand differences in trajectories as opposed to differences in time points, for sure. So we can think of how could that work out statistically, how could that be, interpretable in a reliable manner, for regulatory grade decisions, for sure. I think there's, the big gym for, clinical trial innovation is certainly phase 2 or phase 1B. So a lot of that can happen in that space and then be, confidently taken into phase 3. Obstacles are for sure conservatism and silence. That comes a lot from our stakeholders. I want to say we try to run after innovation as fast as we can, for sure. But when innovation gets inside a company, any company, it creates a period of destabilization. That is understandable. And, everybody's trying to protect a highly regulated environment. That is also understandable. But there's a lot of new technology at the same time and is moving very fast. So from the technology perspective, I'm starting seeing foundation models trained with wearable data. Experimental level at this point, but really fascinating as an option. And I think, we can get there faster than we think and then we need to just try to catch up with all of these and take the opportunities. Another obstacle, I think, is high quality data. Unfortunately, and everything that stands between us and the future is high quality data. We run out of data. So that certainly is something, again, that can be leveraged in early phase development and used and shared. Sharing has to be our goal everywhere. So I was happy to hear Dime saying that they have a repository, of examples of validated frameworks. We have some, but certainly, we would like sponsor not to start from scratch, technology developers not to start from scratch every time, just leverage on existing validation frameworks or existing benchmarks. So that's also another opportunity.
And let me take that opportunity to throw things over to Ben.
All right. Thank you, Christina. So I think if we're talking about the future state, like everyone so far has commented on, if we're in this room, we're probably convinced of the value of a DHT to capture meaningful aspects of health and also improve operational efficiencies across the board. And to Christine's point, we have to act now, or we have to act yesterday, whichever one you want to choose. So instead of repeating what others have said, which I agree with, I'll try to focus more on how I think we should get there. And I think one important driver is that we have to make it a no-brainer for clinical and commercial teams inside a sponsor to entertain incorporating a digital endpoint so that they create the pull to the other teams, to the specialists on DHTs, to actually work with them to make that possible. And again, because the benefits are so tangible around making therapies more patient-centric, starting from a meaningful aspect of health, reducing costs associated with both length of trial, recruitment issues, especially in decentralized settings, all of those things that we all know, right? But it's more about if people who are specialists in DHTs just keep pushing other teams to try and incorporate the technology, it's going to come across as incorporating technology for the sake of incorporating technology as opposed to something they want and need, right? And I think if we can flip that conversation around in making it very clear to those teams and upskilling them, educating them on what this could mean for them so that they create the pull towards the specialists and the experts in the room to help with that implementation, that is going to be a very important driver to make that 5 to 10 year vision a reality. I think that's one component. The second component is that I think we should pay really close attention to those use cases where there currently are no good traditional endpoints. Because what happens then, if you have to fight against an established endpoint, there's going to be a lot of inertia in the room. There's going to be a lot of friction in the room. Whereas there are several examples where that is not the case. And it's going to be, at least in theory, it should be easier there. There should be less friction to show what a digital endpoint can do under those circumstances. And there's two examples, recency bias, that come to mind. One is because, Christine, you were kind of indirectly alluding to it in your opening remarks. If we're looking at the use case around immunotherapies and specifically CAR-Ts and T-cell engager therapies nowadays, they have been, the access to those therapies has been more limited than it could be because of the risk around cytokine release syndrome, right? And that is, so CRS, and it was associated with several FDA REMS programs and so forth, although that's going down now, which is a good sign. But we know that we can improve the clinical management and the way trials are structured for these types of drugs if we have good CRS risk monitoring in place. And Christine referenced the DECODE banner. One of these is DECODE CRS, which Diamond and Ametris are collaborating on to develop resources and implementation studies for exactly that purpose. And I think that's one example where there is no other way to measure this properly. I mean, you could go to self-reports, but we all know that that is nowhere near as accurate as continuous monitoring for this kind of side effect. So I think that's one example. And another one that is very top of mind because we're actively working on it right now is pediatric rare disease, where in many cases it is just prohibitively expensive to develop a true primary endpoint for such a small population. And so work that we have done in the past and that we are continuing to work on now and are kicking off a new iteration is focused on how can we capture functional outcomes that are that apply to more, that translate across more than one rare disease population so that we can start working towards a understanding of what is truly meaningful, what are functional changes that are meaningful to kids and their caregivers in those situations, and how can we use that to connect it to developing better therapies and also developing them much more rapidly than we currently are. And again, just as two examples of how focusing on areas where there really are no good traditional primary endpoints is going to be important to gain that momentum that we need to achieve that 5-year vision. I'm going to say 5 years because 10 years, let's not even talk about that. Let's focus on 5 years.
Fair. Gabriel, do you want to add to that?
For sure. So kind of in Ben's spirit of let's talk about how we get there, I do see a lot of in that 5-year time frame, just through looking through the questions that I've seen in the Q&A panel on this in this session today, I've seen a lot of questions about, for example, what patient meaningfulness data would look like, or questions about what successful CGM applications have looked like, or use cases have looked like. So there's this dynamic where kind of the first one through the glass is going to get a lot of cuts, and it makes it a lot easier for everyone else afterwards. So I see this kind of yearning for like public, concrete examples of successful validation. clinical and analytical, to help others repeat that success. And there are a number of different ways that you could approach trying to provide those public concrete examples to help facilitate more broad success at bringing DHTs into clinical trials, like DHT endpoints into clinical trials. So Christine and Benjamin mentioned Decode. Those sorts of pre-competitive collaborations have in other areas shown a lot of success. And so that's extremely promising. It also seems promising to have some sorts of public-private partnerships. It brings a lot of additional credibility to see the data being real and also being evaluated by the actual validators. My background is in machine learning. You trust the test data a lot more than you trust the training data. So I suspect that when it comes to trying to really convince people, like, yes, I would like to get a DHT endpoint in this clinical trial, having those types of examples to point to would add a lot of additional confidence.
And Gul, I'll have you close us out on this question.
Sure, thank you. Of course, I agree so far what has been done, but maybe briefly add this, the improvements in technologies, advancements is really very impressive. And also integration of this technology in our lives from a patient acceptance point of view. I mean, many people have these wearables now as a watch and etc., monitor their health data. So that's also improves the acceptance part of it from the patients. That's one angle. Another point maybe to think still, I mean, we have guidance documents, what is needed for regulatory acceptance, but additional clarity. How do we get there? Because it's not like always technology is ready for the prime time. We take it and think about all these things and go to the registrational study. It just usually doesn't happen like that. We start initially maybe have it as an exploratory endpoint or in early studies, test out like the pilot studies previously mentioned, maybe slightly different, but have some early ideas. And I wonder if something internally we use, but having a more a framework that we have input from health authorities and alignment from manufacturers and other industry device manufacturers I'm referring, and also other industry input, having a tiered evidentiary framework. What would be the absolutely necessary for using exploratory endpoint? What is absolutely necessary for minimum requirements for secondary endpoint? And then we come to primary endpoint. Having that align this endpoint hierarchy and regulatory decision-making expectations, and having input from the manufacturers, industry, and health authorities alignment, like maybe Benjamin was also previously mentioning. So that could be potentially helpful. And the more we have these standards, one of the vision is also these technologies will be used also in clinical practice. And this will also help us more standardize real-world evidence collection. And that could also support the drug development. I know it's a different pathway, but also overall acceptance point of view. Thank you.
Thank you. I'm going to jump right off of something that you spoke about right at the top of your answer, which is really around this idea of additional guidance and standardization. And several of y'all touched on this on your points. What would you say is most urgently needed? What kind of guidance and standardization is most urgently needed to really achieve that impactful improvement? And maybe taking a little bit of idea of looking at the continuous glucose monitor tech specs. Has that been helpful, and can that be applied more broadly? Are there other tech specs that would be useful? And then also, and this is always a problem in the AI and the digital health space, definitions. We really see that all the different stakeholders often use the same terms to mean different things, which can really create a lot of confusion and then delays because of that confusion. Are there areas where we see inconsistent terminology being particularly problematic? Is that a space that also maybe needs some guidance or clarification as well? Christine, can I start with you on this?
Yeah, happy to. I think that, maybe just to kind of think about terminology. I actually think that we're making good progress in clarifying terminology. Certainly, it's still for people who are paying attention to the guidance. Again, we're going to talk about the white paper that just got released. I think we are seeing, a new term, digitally derived measure, I think. So I think those has been quite helpful. But we still see confusions between like a biomarker and outcome measures. I think the white paper again talk about the difference. So I would encourage everyone to really review that. It's like Valentina said, it's contains like the core knowledge you would need to think about building a DHT strategy. And I also like that the Dr. Fisher earlier talk about another confusion that I've seen is sort of the device clearance versus endpoint validity. Those are two different issues and going to two different FDA review divisions or FDA department. And in our sort of conversation with sponsors, a lot of times people kind of get confused. Is a device clearance guaranteeing your endpoint validation or vice versa, right? So that's something that I think people need to kind of understand. I think the parts that tend to get stuck still with the terminology is this clinical meaningfulness. I think there are ways to get it, but there are challenges I feel because clinical meaningfulness, a lot of times people tend to think about the existing standard. And if we continue to think about how we anchor the new measure to an existing measure, we're actually not moving forward. And then certainly patient centricity is quite important for that meaningfulness. But I think also earlier on, there are several speakers talk about that there are sort of data-driven ways that would allow us to get most out of the digital data. And that might not be the most intuitive things for patient to kind of comment on like what the metric actually means, right? So how can we think about really leveraging the technology in a way that provide the maximum benefits to patients and clinical development and evolving perhaps the clinical meaningfulness to the new generation of technology. So yeah, I'll just comment on that. Yeah.
And Gabriel, I saw you nodding along, so I'm going to throw it to you for a minute.
Yeah, so one thing that's worth highlighting there, I think when we're talking about patient meaningfulness, it will inherently vary. It's not a single analysis. And people would like to boil it down to a simple repeatable framework that can just get copied and pasted across many different indication areas. That's not a very realistic wish. But you mentioned earlier the CGM tech spec. I do think that has moved us forward. I have been able to very practically respond to questions by saying, look at section 3.2.3.2 and like ask me why I know that that's the section that refers to missing data in atom level analysis data, right? It's because it's important. So absolutely, I think that more guidance in that vein, possibly for different modalities, could help in a similar way in moving the ball forward and using DHTs more practically.
Cool. Do you want to add on to that?
Christina, may I jump in?
Oh, please.
Sure. Just wanted to follow up on what Gabriel mentioned. I think, of course, creating more standardization with tech specs like the CGM one is definitely very critical and important. It requires a little bit of maturity of the technology to be able to sort of like speak about how you can standardize something like this across the field, but definitely very, very needed. And I think when you think about this, you feel that there's like there's the poetry and there's the plumbing. So you have something that's like very high level, but you also need the mechanics of things, and the tech specs are very critical to actually get the real work done and have a very efficient process, especially from the regulatory perspective. But I wanted to touch upon something that Ben expanded on, which is I think where we perhaps need guidance, and this is going to be difficult work. It's not going to be easy, but I think it doesn't mean that we don't have to do this work, but areas where we don't actually have strong anchors, because in most of our discussion, we speak about how you're going to compare your measure or the event that you are interested in to an existing biomarker or measure. But when this is not here, this is also a challenge. But when you actually look at the ecosystem and you look at the areas where there's real unmet need, this is where you can actually make a huge impact. When there's a void, or you don't have an existent anchor or a biomarker or a measure that can be used, then where can these drug development programs go? And I think this is really what we should focus in the future on. It's not easy, but doesn't mean that we should stay away from something like this. And I think this is something that we can put some energy in the future, and focus on, of course, in addition to providing more clarity on standardization and providing more tech specs. Because I think when you think about it at a high level, you don't really need to use DHTs just for the sake of using DHTs. You have to ask you the question here is like, why do I need a DHT? And I don't think there's a better incentive or reason when you don't actually have a real measure or a real endpoint or a real biomarker that you can rely on. This is when you can really fill an important gap that we currently see. And Valentina spoke about this in her field. There's a lot of areas that need work, and this is where perhaps digitally derived endpoints and measures can fill this gap.
Oh, did you want to add on to that?
Sorry, is it to me?
Yes.
Oh,
Sorry.
thank you. Sorry, I couldn't catch it. I think new guidance is certain areas is important, but also I would like to emphasize that examples illustrating acceptable approaches, common challenges, and evidentiary expectations will be quite helpful. Maybe one area further providing details will be, I mentioned in my opening statement, particularly challenges associated with passive recording, wearables type of thing, and the influence of external factors. How do we manage this? Is it the patient diaries? But patient diaries also have their own difficulties. Compliance not easy. So I think getting some detailed guidance for that will be helpful. But I would like to emphasize that examples are going to be very, very helpful, with detailed examples. With the white paper, there are nice examples putting the verification and analytical and clinical validation into context, but a little bit more granularity, all those examples, particularly challenges, how they can successfully resolve will be very helpful. Thank you.
And Valentina.
Yes, so I can speak to that a little bit. You heard this morning that we put out a higher request for information.
I'm sorry, Valentina, you're cutting out just a little bit again. If you come in a little bit closer, I think we can hear you better. Yeah.
Yeah.
Perfect.
I was saying that we have an RFI out that closed just June 1st. You heard a little bit of this morning, and I echo that one of the feedbacks that we're receiving is to try to have examples, in fact, shared. So there's different ways that we can do that. We can publish more, but also we could have a repository of shared evidence that couples validation journey with context of use, because I think that is, if I have to pick like one thing that one gets really confused about is really defining the context of use. We have a lot of meetings on this, and I think that that's going to help, like to try to pull side by side, you know, validation level and context of use. Now, I will echo what Gabriel said. You cannot copy and paste from examples because of the nature of clinical validation. Clinical validation isn't the relevant population, precisely that you're enrolling in that trial. And even if there are areas of unmet need, you can isolate a measure and say that even though the measure is accurate, it means something for the patient. So there needs to be some qualitative work around this. But again, there are some examples that we haven't shared that we can share, and we can have for public you know, usage. And I think this is potentially something that we're discussing either as a publication or something similar because of that problem that is coming up. There's a lot of confusion, I think, in what validation means and what qualification means. Again, qualifications a regulatory pathway that's voluntary. You don't have to get into qualification. Validation, on the other hand, is a scientific need, is what really tells you whether the measure is accurate and whether it's clinically meaningful in the population of interest. That is a scientific quest for accuracy as opposed to a regulatory one. So I think that all of that framework needs to be put out there with more clarity, you know, because I think clarity is given by example, if this is what I'm hearing.
And Ben, I'm going to end with you for this question because it sounds like you spent some time collating what exists already. So I'd love to hear where you think there are some gaps.
True. I also know that we're getting close on time, so I'm going to try to keep this as brief as possible. So I think on your first question on the additional guidance, maybe a bit of a, I don't mean it for it to be a flippant take, but maybe we don't need more guidance right now, but we do need very clear use cases of what works and what doesn't work. And I think others have touched on that. I would say one thing though, which is that I think the CGM case and the tech spec around it is a great example of what this could look like. However, because it's a bioanalyte in the end and not a pure physiological behavioral measure, it is different. And I think we should be careful not to try and take what happened there and just kind of match it on the broader category of different modalities that can be captured with digital health technologies, because I think that would backfire in terms of leaving the flexibility to actually define meaningful aspects of health. And then on the second question on kind of terminology and confusion around that, I would say that from our perspective, the terminology issue is solved in one regard, which is that I think we have the proper definitions. Obviously, there's always exceptions, there's always new things that come out, and we always need to continue to work on that. But the foundation that we have of terminology and definitions, I think, solves the need that we have. What is lacking, though, is training and upskilling, making sure that people actually understand the definitions we currently have so that we can have conversations where we're all saying the same thing, as opposed to what, to everybody's point, still happens a lot today now.
Thanks. All right, I have been given a extra 5 minutes so that we can do a quick round robin. I'm going to be asking all of y'all to spend just 30 seconds to answer this, but I do want you to think, kind of given everything you've heard in this meeting today and in this panel today, I'd love to hear from each of you, what is the most important next step for advancing the use of these emerging technologies in a really scientifically rigorous way? And Valentina, I'm going to start with you.
Speak on behalf of regulatory agencies, I'll say shared evidence repository for digital measures with standardized validation evidence and a clearly defined context of use is one thing. As I mentioned initially, alignment potentially across agencies, and another thing is facilitate a pathway for questions, so that every time a sponsor has a question that is not directly related to a development program and doesn't want to enter qualification, we need to have a space where that question can be asked.
Christine, I'm going to go to you next.
Take advantage of the guidance as to Ben's point. I think that we actually have provided a lot of clarifications and frameworks. So take advantage of those, and we are here as partners. So, we're here to support you on this journey. Don't be afraid. And I think that we're here to help you leverage the data that you can't really get out of the trial, that the patient spend effort, and you also the sponsor spend lots of money to do. And I think DHT, you can really help leveraging the effort that we're making to make the most out of the trial data.
Gabriel?
Yeah, so the question is, what's the most important next step?
What's the most important next step?
And here is my claim, in certainly less than 30 seconds. Transparent, traceable, justifiable data flow.
Wonderful. Ben?
I'm going to repeat one thing that I've said in my intro because I think it's really critically important. We need to collectively focus on those use cases that have no good traditional measures available right now, so that we don't have to fight against the inertia associated with these established measures for the right or the wrong reasons. And so that we can simply focus on identifying meaningful aspects of health that are digitally measurable and ground them in the evaluation of the medical product that it's intended to improve the life of that patient for. No good traditional measures. That's where we need to focus.
And Hussein?
I would say perhaps the most important thing, at least from my perspective, is actually thinking about when the conversation is going to start and who's going to be in the room when this conversation is happening. I think timing is very critical. I know that there are challenges with some of the terminology, some of the definitions. There's always going to be questions about what do you mean by meaningful aspect? How can you translate this? And I think if you're able to bring the people that are needed for this discussion at the right time, before you put your validation package together, along with the regulator in the room, and I know that there is a need for additional pathways to do this. As Valentina mentioned, we have to provide other pathways to let sponsors and developers come and speak with us. I think this will definitely help the field to move forward. If we get this right, we can perhaps also address the rest of the challenges as well.
Wonderful. And we're going to let you have the last word, girl.
Okay, so 3 things very quickly. More examples, case studies with data sharing. And also, I again highlight this global alignment is going to be essential going forward. Thank you.
Thank you all. This has been an amazing panel. I really appreciate y'all all joining us for this wonderful session. And I am going to hand things over to Mathilde Kamm for her closing remarks.
Okay, thank you, Christina. Thank you, Christina. Good afternoon, everyone. On behalf of the FDA and our partners at the Duke-Margolis Institute of Health Policy, I want to thank each of you for your time, your expertise, and your engagement throughout today's workshop. So as we close out the afternoon, I want to take a few minutes to reflect on what we've heard, what it means for the field, and where do we go from here. So this workshop was organized around a simple but hard to answer question. That is, how do we develop, validate, analyze, and interpret digitally derived endpoints in ways that are rigorous enough to support regulatory decision making? So when we set out to convene this event, so our goal was to bring together the right voices to answer this question. So we brought together regulators, statisticians, clinicians, industry representatives, and patient advocates for an honest, rigorous conversation about where we are and where we need to go. So looking back at the day that we had, I think we've accomplished that, but more than checking a box, I think we've been able to surface something useful. We now have a clearer picture of the shared challenges that remain and a stronger sense of the collaborative work that is required from us to address them. So we began this morning with an important grounding in FDA's ongoing regulatory efforts and future plans under the PDUFA VII. We had the landscape analysis of recent DHT submissions that made clear that the field is moving quickly and that our regulatory frameworks must keep pace. The patient perspective offered by our colleague from the Michael J. Fox Foundation was a critical reminder that the ultimate measure of these endpoints is whether they capture something real and meaningful from the patient's lived experience. So our second session took a deep dive into the continuous glucose monitoring as a model for how the field can mature. So the CGM tech spec represents exactly the kind of concrete, actionable guidance that sponsors, developers, reviewers need. So it demonstrates that when we work together around shared data standards and analytical best practices, we can move forward from promise to practice. So the CGM experience offers a replicable template for the other DHT modalities. In sessions 3 and 4, we got into the harder statistical and methodological questions. So Badim talked about the concept of data provenance, understanding where the data comes from, how it was processed, and what assumptions have been made before it even reaches an analyst. Remote monitoring introduces gaps, artifacts, dependencies that traditional trials data simply does not have, and our statistical frameworks must account for that. The discussion of analytical and clinical validation, both from regulatory and industry perspective, highlighted a shared challenge. So these two forms of validation answers different questions, yet both are essential. Neither on its own is sufficient to establish confidence in a digitally derived measure. We also heard from Mitch about the importance of distinguishing between validating known endpoints versus developing entirely new endpoints, noting that the latter requires a much higher validation bar. In session 4, we examined one of the most persistent challenges in analyzing digitally derived endpoints, that is missing data. So missingness in DHT trials is often informative. As Cynthia mentioned, it could reflect device failure, patient burden, or disease progression. The panel discussion also dealt with a difficult but important threshold question. That is, when is there still enough data to trust the results, and when has missingness gone too far for any analytical method to reliably impute the missing data? These are not merely technical problems. They are questions about the integrity and interpretability of the evidence we use to make regulatory decisions that affects patient lives. So the presentation and panel discussion today both surface both the complexities of these challenges and the genuine progress that is being made to address them. And in our final session, we looked ahead. The future of digitally derived endpoints is not in a distant horizon. It is here now. So in the form of novel sensors, passive monitoring technologies, machine learning derived features, and endpoints that captures dimensions of patient experience that traditional clinical assessments simply cannot reach. So the question before us is not whether these tools will play a role in drug and biological product development. They already do. The question is whether can we build the scientific and regulatory infrastructure to support their use with the rigor and transparency needed for regulatory decisions. The panel also emphasized the need to focus on use cases that don't currently have good traditional endpoints. They mentioned that this will help highlight the benefits of digitally derived endpoints. So as I reflect on today's discussion, I've come up with a few cross-cutting themes that stood out that I believe will shape our work going forward. So the first is standardization is fundamental. So the conversations today about data submission standards, terminology, alignment, and analytical conventions are conversations that require ongoing collaboration across all stakeholders. So it has been mentioned that inconsistent terminology, so a point that was raised in session 5, is not just a communication inconvenience, it creates real barriers to cross-trial comparability and regulatory review efficiency. So getting aligned on the language is fundamental to everything else. So second, the statistical methodology must evolve alongside the technology. So DHTs generate data that is fundamentally different in structure, volume, and complexity from what traditional clinical trial endpoints produce. So the standards we apply to the digitally derived endpoints must be as high as those we apply to any clinical endpoint intended to support a labeling claim. So it's the same. It doesn't matter what endpoint it is, whether it's traditional or digitally derived. We should have apply the same standards. Our analytical methods need to reflect that reality. So we heard today about promising approaches to handling missing data and validating new novel endpoints. And there is important work still to be done, and FDA is committed to engaging with the broader scientific community to advance these methods. Third, I would say you've heard it across the 5 session. Patient perspective must remain central. So digitally derived endpoints only matter if they measure something that matters to patients. The integration of patient input into endpoint development from concept elicitation through the labeling claims is not optional. This was said in session 1, but it echoes throughout the day. So a digitally derived endpoint that is acceptable to regulators but not meaningful to patients, or that places an acceptable burden to the patients, is not fit for purpose. And fourth, early and iterative engagement with the FDA matters. So the gaps and challenges we've discussed today in data quality, validation, missing data, analytical decision making are far easier to address before data collection begins than after. FDA's programs for early engagement exist precisely for this purpose, and I encourage the sponsors to use them. So today's workshop is part of a broader and sustained effort. So FDA's PDUFA VII commitments reflect a recognition that advancing the use of DHTs in drug development requires not just guidance documents, but ongoing dialogue, iterative learning, and genuine collaboration across the different sectors. So we will be carefully reviewing the input shared today, including comments submitted through the public docket as we continue to develop our thinking on these issues. So we also recognize that there is more work to do, and FDA is committed to unlocking the full potential of DHTs to support medical product development and innovation. So the opportunities associated with DHTs align directly with the FDA's mission to protect, promote, and advance public health, and they build on the agency's long-standing commitment to supporting responsible innovation in the development and regulation of medical products. So I want to close with a word of genuine thanks. So to my FDA colleagues from the Office of Biostatistics and the Office of Medical Policy, thank you for your assistance with planning, and organizing today's workshop. To our colleagues at the Duke-Margolis Institute of Health Policy, Cameron Joyce, Alisa Wong, Val Parker, as well as Hannah Vitello and Luke Doshert. Thank you for your partnership in convening this workshop and for your continued commitment to advancing evidence-based health policy. So to our speakers and panelists from FDA CDER, CDRH, CBER, and OB, as well as from Johns Hopkins, the University of Michigan, Stanford University, and industry. Thank you for the depth and candor of your contributions. And to every participant who joined us today, asked a question, or submitted a comment, your engagement is what makes this process meaningful. I know we were not able to address all the questions in the Q&A today, but we will read them and think about them, and we appreciate your engagement and thoughtful discussion. So the work of developing rigorous, patient-centered, scientifically sound, digitally derived endpoints is not easy, as you've heard today, but it is important, and today reminded us that we are not doing it alone. So thank you, everybody, and we appreciate your attendance.